Good morning, everyone. Hello to all of those of you in the room. For those of you who are online, we anticipate about 95 people to be in person over today and the next 2 days. And for those of you in the room, there are 2,600 people registered online. So my name is Jim Polli, and welcome to the Bioequivalence Innovations for Generic Oral Products, Biowaivers, Bridging, and Development for Oncology and Discontinued Products. So this is a 2-day CRCG workshop on BE. And for me, it's kind of an unusual BE meeting. I've been to many BE meetings over the years, and this one's really nicely focused on oral problem issues. And so I really look forward to the next 2 days. Okay, just a little bit about the Center for Research on Complex Generics, which is hosting this meeting. It's established in 2020 by the FDA. The CRCG is a collaboration between the University of Maryland and the University of Michigan, along with, of course, the FDA. And the mission is to increase access to safe and effective generic drugs through enhanced infrastructure and communication, education, and research collaboration across industry, academia, and FDA. I'm happy to say there's a lot of FDA folks actually here in the room who are very interested in bioequivalence. And the primary goals of the center are infrastructure and communication, education and training, and collaborative research. And before I forget, I was just going to say to everyone in the room and folks online that these slides, as well as really all the slides, will be available within the next 3 weeks, as well as the recordings. If you're online, you're seeing a recording right now. And we will email you when they become available. And they'll be posted on the complexgenerics.org website. So one element of the center that we're sort of mandated to do with the FDA, a requirement, is to have ongoing engagement to advance complex generic product development. Much thanks to many of you in the room and those online with whom we've already spoken with over the years. And I will say over the next, we've taken a little bit of a pause over the last few months, like six months. And now we're going to, for the rest of this year, 2026, we're going to have a renewed effort as far as outreach and asking questions about, what research should be done to advance complex generics and this sort of thing. So if that's something you're interested in speaking with the center about, Center for Research on Complex Generics, feel free to just send us an email right now. Just a little reminder about upcoming workshops for the rest of the year. We typically have 4 a year. This is actually the first one of 2026. And we've got 3 workshops in the fall, in September, October, and December. They'll be in this room. They also, of course, will be online, although it's better in person, I'll say, particularly because of the small group discussions as we're having at this meeting. In September, navigating the GLP-1 generic pathway. In October, it's on inhalation nasal products as far as using in vitro, in vivo, and in silico methods to assess bioequivalence. And then before the end of the year in December, a workshop on long-acting injectable generics. So we do a lot of outreach and communications and workshops and that sort of thing. We also have research projects. This is sort of a list over the last 6 years or so. I'll just mention a couple that I think are relevant for this meeting. Towards the bottom there, you see some references to nitrosamines and antioxidants and that sort of thing. And towards the top there, we did have some projects early on looking at long-acting injectables. So this is the CRCG team. Again, my name is James Polli, and Schwenneman is here, also a co-director. Dana is also here. For those of you online, frequently interact with her. She's actually here in person. And Visha and Jennifer are online. Okay, here's just some information. Please sign up to receive our emails. We send probably about one a month, I must admit, usually related to workshop to keep people up to date about that. Again, just huge thank you. This was a workshop probably it started after its conception, probably about 6 months ago. So a lot of people on the FDA side and also on the center and industry side, I guess just Siva, Emilija, and Tausif folks representing industry, just special thanks for them. Again, thank you for your participation, for coming here. I know some people came from across the globe to get here, and those of you online are maybe staying up kind of late already as the day here in the U.S. progresses. So thanks for your participation. Just some reminders that we do have panel sessions at this meeting, so we'll be taking questions from the speakers from the audience, both here in the room and as well as online at that time during the panel discussion and not after every talk. Everything's being recorded, so this is a public meeting, which does mean, you know, if you if you ask a question, we will be recording that. And again, just a reminder, if you can always check complexgenerics.org and within 3 weeks, although with regard to availability of materials for this workshop being online, although we'll also email all registrants. Okay, so it's my pleasure to introduce someone I think all the folks online probably know, Rob Lionberger. Rob is the director of ORS and OGD, and you know, I've introduced Rob before, and I'm always struck by the role of the FDA OGD in pushing public science such that products, including of course complex products, can be assessed. And I can't tell you, Rob, the number of people that comment about how they're very grateful. These are people overseas. They're very grateful for FDA's leadership on pushing the product understanding science. Okay, Rob, thanks.
All right, thank you, Jim, and welcome to everyone who's here and everyone online. Again, like Jim, I want to emphasize, you know, when we have these CRG workshops, there's huge value add for you if you're able to attend in person. This is really an opportunity to have, you know, through our sessions, but also through the informal discussions, interactions with the key FDA staff working in these areas, as well as the key people from other organizations who are working on this area. So we really encourage you as you look at the schedule coming up to really, if you're working actively in, you know, novel, you know, in this case, novel bioequivalence approaches, or if you're working on peptides or you're interested in a generic company that's interested in making GLP-1 peptides, we really want you to be here in person for that meeting. We can hear from you, your feedback, that's really valuable to us as FDA, to have that direct feedback of what are your real experiences in working with and developing these products. That's immensely valuable to us and to you. So we hope you look at the CRGC schedule, look at those upcoming events, and if you or your company are actively involved in developing those products, really plan to have someone from your organization here in person for that interaction. You know, we're very proud of Jim said, the wide range this event gets through the broadcast media, but there's additional value that gets added in, internally in the in-person event. So I really want to emphasize that. And I look forward to having those discussions with all of you who are here today. And so for my introduction, I want to share a little bit about what I think is necessary for bioequivalence innovations. And as we think about this, maybe I'm going to say some things that are a little bit controversial, but I think we have to move beyond the BCS thinking to something I want to call quantitative biopharmaceutics. And I'll try to sort of just briefly explain that in this. And again, this is really an opinion talk. This is not an official FDA statement. These are my thoughts about where the future of this field is going and what we're going to have. And I think that as we start talking about how do we have innovative approaches to bioequivalence, i.e. ones that don't necessarily require the traditional in vivo studies, we have to start with the biopharmaceutics classification system, which, even though I'm going to be critical of it, has had fantastic successes, right? It's led to a way of thinking about BE studies. It's led to waivers for certain classes of products. But as we think about looking beyond where we are with the DCS guidances and into places that are more challenging and more impactful for access to affordable generic medications, we have to think about some of the the challenges that have come from the 30 years of the BCS to move forward. And like the theme of my talk is I think that in the future, innovation is really going to come from the intersection of what I call quantitative medicine. That's predictive models, mechanistic models of drug absorption, and AI-empowered data sources and model development tools. This is going to be how oral drug availability, pharmaceutical science is really done in the future. And in ORS, we like to refer to this as quantitative biopharmaceutics. And that's sort of our vision and what we think the direction of the future is. And I want to explain a little bit about where that comes from in my introduction here and take a few minutes to set that frame, because this thinking will be really important to all the topics we talk about today. When you say, like, look, I want to have alternative approaches for oncology products. A lot of them are going to be lower solubility drugs. So we have to be open to new approaches there. So to give you a sense, so I asked, like everyone else, I asked our AI assistant to draw me a little chart of the BCS system. Okay, and I want to say this is reflecting the conventional wisdom. So on this chart, right, it drew a nice little 4-box view, and all of those 4 boxes are equal and they're nicely balanced. And we've divided up the world into 4 sort of equally sized pieces, right? I think this is the first sort of conventional wisdom. But I think the reality of the BCS system is that really and FDA rightfully, almost all of the attention in the BCS at the beginning was focused on class 1 products. We can give them waivers, right? But then FDA said, well, if we're going to give waivers of these in vivo studies, we have to be pretty conservative and make sure that these products are going to do. So in effect, it sort of started squeezing that class 1 box to be very much smaller, right? Really only really high solubility drugs and really highly permeable drugs that we really know. But the consequence of that is, right, every time, if you think of the big X on this, every time you make class 1 smaller, you make class 4 bigger. And that's sort of, I would almost say, an unintended consequence of thinking about things through the BCS system and its intersection with FDA's regulatory processes. And there's been some implications of this that, again, as you start saying, like, well, now I want to have an alternative B approach for something that's not in the class 1 drug. Is everything else high risk? And so when I really want to, like, at FDA scare people, say, like, are class 4 drugs high risk? And I want to make people say, like, class 4 drugs aren't high risk. And then people really get nervous about that. And then they say, well, maybe say, like, class 4 drugs aren't always high risk is a way to think about that. And part of that is because when you've been cautious about class 1, you suddenly make class 4 bigger and bigger and bigger. And there are a lot of things in there that aren't necessarily high risk. And I think that's the biggest thing I want people to think about is that there's this unintended consequence of how we've thought about BCS over the many years is that the other classes, not class 1, contain a mixture of higher and lower risk products. Like that's the conceptual idea is, and as you start applying more scientific approaches and looking for products where alternative approaches might be feasible, really sort of understanding that is, why is this product in class 4 or class 3? What elements of that make that higher or lower risk within those other BCS classes? And that's a really important, like, conceptual breakthrough that you have to make in order to move forward. And like, so give another example of this. So because you've made, class 1 so small, you've made class 3 so big. Right. And if you look at the M9 guidance, it's actually pretty cautious, right? Because it's trying to have one approach that works for almost 0 absorbed product and an 84% product. All of those are in class 3. There's a wide variety there. If you have one size fits all for those, you have to be more cautious. And so you can look at different approaches there. Again, one thing I really like about M9 is that it introduced the concept to the BCS of looking at formulation similarity, right? So, M9 says, the formulations have to be very similar. Maybe it's being too cautious about that because it has to cover the whole thing, but at least it introduced that concept. And one thing I want you to think about at this workshop is when you go to the low solubility side of the BCS system, what's the role of formulation similarity for managing risks there? In other products, non-oral products, ORS and complex generics have had a lot of success in thinking about what we call Q3 approaches, not just Q1, Q2, but characterizing the physical state of the products, right? So perhaps a product that's very similar to its reference product in Q1, Q2, and Q3, and Q3 might be, maybe I'm making a solid dispersion. Maybe I have an amorphous dispersion, and I can characterize that, and they can be very similar, and you can measure that with analytical tools. Like, again, that's a conceptual framework that I think has been missing, but is an important part of this. So second point of BCS gap is, right, if you ask your friends who are biostatisticians, should I take my continuous variable and reduce it to a categorical variable and then do my statistical analysis? They'll probably say, no, don't do that. You're just losing information. So again, every time we sort of take something and sort of say, well, I'm going to reduce this to class 4, class 3, when I really have continuous variables of the solubility. Like, I know what the solubility is. The permeability or fraction absorbed, maybe I also have more continuous measures of those things. So we lose valuable information when we reduce things to categorical variables. And so if we want to have a better risk model that applies more broadly to the products people are interested in developing generic drugs for, we don't want to lose that information. We want to regain that. And that means thinking more continuously. The other part of the BCS classification, and this is something certainly in the literature you can read about, but in the simplest classification, it ignores weak acid and weak bases, which all have low solubility, all get classified as low solubility. But those products have oftentimes high solubility absorption windows. There are many successful pharmaceutical products that are successful because they are weak acids or weak bases, and they have an area in the GI tract where they can be readily absorbed. And so if you say class 4, but it's a weak acid or a weak base, that could be very different from something that's low solubility in all conditions. And if you don't understand that, again, that's the place where by changing to categorical variables, you're throwing away information that is relevant to thinking about risk more clearly and precisely. And I think that's what we want people to be open to today and in the future as we talk about the different applications and places where innovative bioequivalent approaches are important. So how to move forward? Like, this is the way I think the future will look. And this is what we want to call, what I call quantitative biopharmaceutics. Again, we're using more quantitative medicine, predictive models that take as input the continuous and pH-dependent solubility measurements and estimates of the permeability. And again, if any of you have worked in trying to have predictive models for OR absorption, you know that you have to have some input about the product dissolution and release rate, right? And so we'll definitely have a session here today about more biopredictive methods and putting in the, you put those together, that's the right way to think about, are there risks in this formulation? And in ORS, I think we're doing some really exciting regulatory science work relevant to the generic program around this on, I think, a fantastic and innovative scale. And the connection to the AI tools is, for many years, people said, oh, FDA, you know a lot about BCS, you see all these applications. But the reality was, like, no one at FDA saw all those applications. The reviewer just saw the ones that they looked at. People could go through and dig out a few examples if they had a lot of time, but no one had the view of everything. Now, and that was limited by, well, you let me, give me 100, let me hire 100 people and I'll go dig out all the BCS information that FDA has. Now we have AI tools. We can go through and tell them and train them to go look at everything. Right? And so we can really digest all of that. And we're really, we're building a fully comprehensive understanding of the quantitative biopharmaceutics data that's available. Again, also at FDA, we have a lot of PK studies. In the past, when people wanted to analyze PK studies, they could go dig it out of a file and reformat the data, and then they could use that study. We've actually been able to automate that process using AI tools. We have what we call a PK data warehouse. We have access to 10 to 20,000 PK studies, and we're going to digest everything. And so we can then compare, I want to compare my quantitative biopharmaceutics predictions to PK data. We can do that on a scale that's never been done before. And again, AI can also accelerate the model development. We're really interested in how we do biopharmaceutics modeling at scale, not one product at a time, but in parallel, do 1,000 products at a time. the compute limitation isn't there, right? We have enough compute power. We're spending all of our compute power on AI tokens. One small fraction of that can build quantitative biopharmaceutic models and solve differential equations. So we can do that at a bigger scale. We can use AI tools to draw on this data. So this is a fantastic opportunity and point. There's no other place that you can do this, right? None of our, the big AI companies can do this. All of this data is inside FDA. It's proprietary. It's never going to leave FDA. So you can't go outside and do this. If you want to work on this problem and make a huge impact, you have to come to FDA, to ORS to do this. So that's I have a recruitment slide at the end, but I want to mix that in here. So again, if you're a young scientist working in oral drug absorption and you want to leverage this system and build the next generation, you have to come here to do this. And there's no other place that you can do that. And so, we look forward to doing that. And, right now we do have actual openings, both for full-time scientists and new young and fellowships for recent graduates. So again, please reach out to us and engage us. We're really looking for places. This is your opportunity to get in on the ground floor. If you're interested in AI, it's too late. You should have joined OpenAI 10 years ago. But if you're interested in AI for biopharmaceutics, this is the time to get in on the ground floor. This is sort of your moon landing generation, right? This is your Artemis project, your Apollo project. And, it is beneficial for all mankind, right? This information, if FDA were able to distill this in better ways of thinking about bioequivalence, that's beneficial to everyone. Outside of FDA, each pharma company might have their own things, but they're interested in advancing, their own drug products. This is the place to advance the science that's beneficial for everyone. And so, it's a wonderful and fantastic mission. And the CRGC is an incredible part of that and engagement of that, but we really look forward to doing this. And I'm incredibly excited about this. Now, today at this workshop, we're going to talk about, not this sort of big picture behind the scenes details. We're going to really talk about applications. What are some of the most current challenges to the generic industry where innovative bioequivalence approaches can really make a difference in access to products? And, I think this is what we, as Jim mentioned, this is what we targeted these. And I want to, remind you of these opportunities to do these. For oncology products, incredibly important. FDA is super innovative at approving new oncology products very quickly, right? But again, if you think about how do you have access, for the ones that are successful, that become part of the standard treatment arrangement, how do you provide affordable access to these products at very large scales? And, we've done, you'll hear some studies about these where we looked at the generic access to oncology products lags other therapeutic areas. And one of the challenges is for many of the products, healthy subject studies are not possible. And this means the bio waivers are much more important. We've already gone through the low-hanging fruit. So if there's a BCS class 1 drug for an oncology product, we have a product-specific guidance that tells you you can use that waiver. And again, I think we've also probably captured all of the BCS 3 products in that. But again, if many of the oncology products are lower solubility. And so you have to deal with some of the challenges I talked before and say, which of these products, where are there possibilities for waivers? Maybe if you integrate, maybe you can be more open to approaches if you're able to do Q1, Q2, Q3 comparisons. If you have better biopredictive methods, you have to have some kind of innovation, but those are places where you might be able to do that. Again, nitrosamine is another example. This is one where, FDA's guidance basically says class 4 products are high risk. And again, we've made class 4 very big. So a lot of the products, we want you to reformulate your products to reduce nitrosamines, right? You shouldn't have genotoxic impurities in your pharmaceutical products. Nobody wants that. But we also recognize we have to maintain access and supply to these products. So efficient reformulation. So again, FDA's guidance says, there's shortcut paths for everything except class 4. We'll talk about that here. What about the class 4 products? What approaches are open there? How can you use modeling and simulation approaches to more efficiently reformulate nitrosamine products? And the third, big area is talking about unavailable reference products. So again, FDA went through the trouble of approving a new drug application. We validated the safety and efficacy, labeling is appropriate, but for whatever reason, maybe the brand company exits that market. a lot of times there's already, if a generic is approved early enough, then that generic becomes the reference standard and competition for that product for continued. But sometimes, for whatever reason, like the brand product leaves the market before a generic becomes available, and now you have to make a connection to bring that back. It's just wasteful to the sort of healthcare and pharmaceutical system as a whole. There are, I think, 200 to 300 products that fall into this category right now in our PSG database, where we have a PSG that tells you what study to do, that you cannot do the study because there is no reference standard and there's no RLD available. But FDA went through the trouble. So somebody did a clinical study. Somebody got a label approved. FDA, those products aren't withdrawn for safety or efficacy. They're just not fitting into some big pharma company's plan. If you in the generic industry see opportunities in there and you want to bring those products back, right? Your challenge is, well, I can't follow what's in the PSG because that reference standard isn't there. I have to do something else. And so we'll have a session talking about, well, what are the other bridging things you can do? And putting together, it could be biowaivers, it could be connecting to some other product that has a relative bioavailability that we can estimate. These are opportunities, to leverage the decisions that FDA has made in the past about what products are safe and effective, not repeat unnecessary clinical studies to bring those back. So, all of these are opportunities where innovative thinking about bioequivalence and different approaches can lead to, again, more access to generic versions of these products in areas where there's not currently generic competition. And that's what we've really focused on here. And my advice to you in the industry is, if you want to propose innovative methods, focus on these problems where the innovative method is necessary to provide access to those products. That's when you'll get, I think, better interaction with FDA, more openness to innovation. And we'll have a whole session at the end of this workshop on some of the, because we recognize something FDA says innovation is approach, but when I come to FDA with something innovative, they just tell me I can't do it. So we want to hear about that. And again, but I think here from this workshop, really picking problems where the innovation is necessary to provide access to these products in an affordable way. That's where you're going to get more traction, more interaction, more positive discussion from FDA than saying, well, I just don't want to do a Fed study, right? These are things where the fundamental access to the problem requires new scientific approaches. And I encourage you to engage, across this workshop in this. I think this is a fantastic opportunity for what the generic industry, the generic drug review process is going to look like in the future. And again, the foundation of this is begin thinking beyond BCS. There's something called quantitative biopharmaceutics out there. And I think this will really encourage, and in this workshop, want to encourage innovative approaches for products where the scientific and regulatory challenges limit access. So I look forward to this workshop and engaging with all of you, and hope that all of you also online learn a lot and see new opportunities for your companies to develop and provide access to these products. And again, as I mentioned, here's our slide about our hiring process. So we have an OGD jobs mailbox. You're looking at, not just in my office, but across all the offices of OGD. We have people who are engaged in these process, not just the scientific work, but also the review of the application, the farm tox work that supports that, the clinical work, as well as the regulatory councils that involve in the legal work, our project managers who help organize all of this. Like, there's positions across the range of activities. So again, if you or people might be interested in those opportunities, this mailbox is a great way to contact us. We're currently looking for people who have either U.S. citizenship or green card available in these different fields and areas. And so again, it's a fantastic opportunity to contribute to the mission of this organization, which is fantastic. And that's, I've been here over 20 years and it's fantastic. It's constantly interesting, constantly impactful. And it's a fantastic way to spend your professional career. So with that, look forward to interacting with all of you at this workshop today. Thank you all very much.
Okay, thanks, Rob. Our first session is on our first of 7 sessions over the next 2 days is on bioequivalence approaches for oncology products. We have 2 moderators, Tausif Ahmed and Jihong Song. Tausif is virtual, so unfortunately he wasn't able to be here with us. And so for those of you in the room, we have hard copies of the agenda over the next 2 days. There's also a link on the table as far as where to find bios. For people online, I encourage you to go to complexgenerics.org for the agenda and bios. And, very much appreciate your willingness to read the bios online, since we'll get this brief bios here. But Dr. Ahmed, he's Senior Vice President and Head for Clinical Research and Biopharmaceutics at Mankind. He's one of our moderators. He's someone I will frequently reach out to when I have BE questions. And Jihong from the FDA. He is the Associate Director for Clinical Safety. I'll just maybe say one thing about him. He's the co-chair of the Generic Drug Development Safety Committee. Jihong.
Good morning, everyone. Good morning, good afternoon, good evening, all the virtual attendees. It's such an honor for me to take a moderator role for this oncology session. Dr. Tausif Ahmed from Mankind Pharma is a co-moderator with me. He's joining virtually. He was supposed to give an introductory remark, but he could not present online. On his behalf, let me provide a brief introduction on this session. This session will focus on current and emerging topics related to the approaches for oncology oral product. Today, we will hear from 8 distinguished speakers, 4 representing industry and 4 from FDA. This session will highlight FDA, industry, current thinking and approaches or proposal on the study and alternative approach to overcome obstacles in development of oral oncology product. Following key topics will be covered: ethical and regulatory frame of the study in oncology area, optimization of clinical B study in oncology patients, and alternative approach such as the BCS-based waiver approach and PPK modeling for oncology product. In the interest of time, I apologize that I should skip the introduction of each speaker. You can refer to program brochure for brief bio sketch for each presenter. I'd like to propose that we proceed to the main session topic. Let me introduce the first presenter, Dr. Anuj Kumar Saini. Dr. Saini is a global head global clinical management, Dr. Red Laboratory. His presentation title is Industry Perspective on the Assessment for Oncology Product, Balancing Challenge and Evidence. Dr. Saini is virtually attending.
Good morning, good evening, everyone, and thank you for joining this session. So thank you for giving the opportunity to speak on industry perspective on Bioequivalence assessment for the oncology products. So general disclaimer that opinion expressed in these presentations are solely of myself and doesn't belong to any opinion from Dr. ATs. In the subsequent slide, I'm going to present various challenges as listed here, generic industry faces with respect to clinical development for oncology products. As we know, oncology bioequivalence studies are fundamentally different from other respective areas. They are normally required to be conducted in a patient rather than healthy volunteers, largely due to ethical and safety constraints. These studies are typically steady state, reflecting chronic dosing combination therapy and real-world use. At the same time, oncology patients receive multiple concomitant medication, resulting into high variability of these studies. As a result, bioequivalence assessment in oncology becomes scientifically very complex. Today, I'll highlight key industry challenges and some of the practical solutions of these challenges for further considerations. In the forthcoming slides, I will talk about challenges associated with global development, long-term patient engagement, concomitant therapy, etc. I will also touch base about handling high variability in these studies, recruitment concerns in rare cancer indication, and largely, and challenges related to post-approval changes. Some of the possible solution will also be discussed here in this presentation. Usually, industry aims for global generic development, but current regulatory expectation often require multiple pivotal bioequivalence studies due to differences in reference product, sometimes study population and bioequivalence requirement as well. So this creates a significant execution and a cost burden to the industry, and also ethical concern due to repeated pivotal patient-based studies. A potential solution is leveraging a single PK studies across geographies, supported by a bridging strategy where reference product similarity can be scientifically demonstrated. ICH M13A also offers a possibility of using multiple reference products in a single study. However, it's not always favorable scenario to follow the same. Another rationalized approach is conducting a single study using one RLD and leverage it to multiple geographies. Bridging across a region can be supported through totality of evidence approach. That includes physicochemical characterization, dissolution similarity, evidence of identical compositions, and demonstration that a common clinical trial supported approval across multiple regions. So recently, agency allowed to consider clinical study conducted using non-US RLD for biosimilar application. Also, Health Canada accepts a study conducted using other RLD providing stroke evidences are produced to demonstrate that both the RLDs are same. So with this update, request from industry to agency for similar consideration for oncology and other products. One of very strong evidence is the use of common clinical trial data to secure marketing authorization in multiple geographies. For example, you may see here that the innovator used the data of a common clinical trial, that is SOHO 01, to register its product in US and Canada. So this gives indirect evidence that product is same in both the geographies which innovator is supplying. Most oncology bioequivalence studies require steady state evaluation, leading to prolonged patient participation and higher dropout risk due to disease progression and treatment toxicity. A key opportunity lies in model-informed drug development, including PBPK, population PK approaches. So these approaches can reduce patient numbers, allow sparse sampling, and low patient burden. Regulatory guidance could further expand their use in the oncology area. Concomitant medication are unavoidable in onco patient, as we know, and can significantly have a pharmacokinetic through effect on absorption, metabolism, and elimination. From an industry perspective, early alignment with agency before submission is critical. Scientifically justified handling of outlier where appropriate should also be considered here in this case. This will help in treating the PK data derived from critical patients judiciously. Where repeating the PK study because of outlier is unethical. In order to take care of high intra-subject variability, innovative design, as exampled here in this slide, can also be used based on the pre-alignment with the agencies. Recruitment is challenging in rare cancer, as we know, and biomarker-defined population especially. A clear example is dabrafenib, where treatment is BRAF in BRAF wild-type patients may worsen the disease, making patient-based BE studies ethically unacceptable. In such cases, industry proposes exploring a single-dose animal PK study and then using this data, allometric scaling of this data to extrapolate to humans. This approach merits regulatory consideration where patient studies are not feasible. Handling post-approval challenges is another concern in oncology drug development. Between 2020 to 2024, agency approved 245 new drugs based on the PPPK evidences. In the similar way, it will be helpful to industry that agency comes with a defined guidance on handling post-approval challenges. So to conclude, oncology bioequivalence challenges come primarily from patient-based studies, steady state multiple dose requirement, concomitant medication, and high variability. So these factors can complicate execution, recruitment, and life cycle management. So innovative approaches such as including global acceptance of a single PK study, modeling-based strategies, and oncology-specific guidance can significantly reduce burden without compromising safety. So with this, I acknowledge the help from Dr. Sushil, Dr. Krishna, Dr. Reddy's for providing me all the support, and CRCG and FDA, other industry members for this opportunity and guidance. Thank you so much for your patient listening. Now I would like to invite Dr. Jihong Shon, Associate Director of Clinical Safety at FDA, for the next session. Thank you so much.
Yeah, good morning, everyone. This is my presentation title. I will walk you through current regulatory paradigm and practice that we use when evaluating in vivo B study design for this challenging class of drug. This is my disclaimer. This is a roadmap for my presentation. So challenge for in vivo B study of oncology product. I don't think I need to talk about any particular challenges for development of oncology product because everyone knows well. However, it might be worthwhile to better understand the current paradigm or landscape of these challenges from industry as reflected in the real world experience from developer perspective. So we look into some inquiry in the last year. Left diagram show that 57% of inquiry were related to in vivo B studies. We are the right is a right diagram present breaking down result. This result clearly reveals that a substantial proportion of inquiry pertain to study population and those selection. I'd like to introduce the current agency's approach for study population recommendation. Clinical assessment for study population selection is conducted using multiple resources, non-clinical toxicology profile, safety data from NDA, ANDA, post-marketing data, and public literature. We also look at drug exposure at those intended for B study. We also review all relevant safety profile of other drugs within the same pharmacological class. We also consider pharmacological plausibility for a selected B dose under B conditions. So together, all input form the foundation of study population selection recommendation. So let me give more specific for clinical safety factors which can drive a recommendation on B study in patient rather than healthy subject. Cytotoxicity, genotoxicity, and hallucinogenicity are primary concern if their property pose unacceptable risk in healthy subjects. Reproductive toxicity, such as embryo fetal toxicity and fertility impairment, is another critical factor. We also carefully review all organ-specific toxicity, hepatotoxicity, cardiotoxicity, such as QT prolongation, idiopathic reaction, blood discharge affecting immune function and bleeding risk, and CNS impairment. When one or more component of these factors are anticipated beyond acceptable range, our ethical calculation lean toward patient rather than healthy subject study population recommendation. So from agency historical exercise and recent update practice for study population selection, we had implemented a decision framework approach to make consistency and structure to this decision-making process. So I'd like to walk you through the schematic flowchart, which reflects our current practice. This flowchart operates through a fleet of the checkpoint to evaluate the non-clinical and clinical safety component. If a drug is toxic, we usually recommend patient population. For genotoxicity, we look into more detail of the genotoxic characteristics. Decision can be made using a totality of all the evidence from genotoxic profile with the consideration of exposure limit risk assessment. All safety findings observed in the patient are also considered in healthy subject and the use in the healthy subject with respect to persistence of each identified safety risk and its potential occurrence in healthy subject. So all collective assessment, all these factors can result in the final decision. But I'd like to note that this approach had just captured the main frame for our decision process. There might be more component and consideration that factor into the final regulatory recommendation. So I would like to walk you through our current clinical assessment platform for study population recommendation process. We have built a dedicated clinical assessment review team. This team has used standardized clinical assessment workflow. This is an example of the workflow that we developed. There is an independent and multidisciplinary safety discussion group within OGD. This group brings together experts across discipline to provide, solid, advice on the primary assessment on both study dose, study population, and study design. They want to also ensure that those decisions are supported by best available evidence. I would like to turn to some regulatory advancement in these areas. We had been put in effort with developing the strategy oversight and time action mode on the study population design recently. One key action was the updated process for PSG for oncology product recommending patient B studies. This update can be triggered by multiple factors, developer inquiry, and a new safety data, and a part of the regulatory harmonization and proactive monitoring patient-based PSG like clinical assessment team. Once this trigger factor is captured as determined to be reviewed, the review team conducts a thorough review using the updated information. The potential action from this assessment can include update on study population, patients who are healthy, and a new dose for conducting B study in healthy subjects, and optimization of patient-based B study, including alternative approach. Last year, from this assessment, we updated Bible product to convert study population from patient to healthy. So I'd like to give more detail for this process using one example, lorlatinib. Lorlatinib is approved in 2018. We OGD published initial PSG recommending patient B study based on safety concern of the ECG finding. We also recommended bio-IND due to preclinical toxicology profile. We received inquiry to evaluate feasibility of B study in healthy subjects. We conducted the reassessment using all updated data. We finally conclude that there is a relatively low risk in healthy subject as single dose. We also examined other safety signals, carcinogenicity and hepatotoxicity. There were no major safety signals. So this year, we updated PSG, a single dose B study at 100 milligrams can be conducted in healthy subjects. We also determined that bio-IND is not needed based on the updated toxicology assessment. I think that this example represents how your evidence combined with rigorous review process can reduce burden in genetic oncology development program while maintaining scientific and ethical integrity. I'd like to touch upon some other regulatory advancement. We had been working to optimize the patient B study for oncology product by improving clarity on PSG, such as the food conditions and description of study population for patients. We also try to incorporate the relevant safety measure and exploring alternative approaches. On the regulatory harmonization front, we had actively engaged with other agencies to align approaches and streamline the PSGs. So from this collaborative harmonization effort, we could achieve the alignment on several product listening, and we also identified additional products for the additional products for revision, potential revision. In closing, I'd like to emphasize that the work we do is fundamentally balanced between ethical consideration, scientific aspect. We have committed to making suitable and timely recommendation using best available evidence information in collaboration with the stakeholder as well as experts. We also recognize that the emerging technology is, put on the table. That advancement will be helpful for us to explore new opportunities. So I'd like to take a moment to acknowledge my colleagues whose contribution make this work possible. Thank you for your attention. So let me introduce the next speaker. Dejan Krajcar. Dr. Krajcar is the head of clinical innovation, clinical pharmacology modeling and simulation, Sandoz Global Development. His presenter title is Optimization the Trial on Oncology.
Good morning, everyone who come here today, and good afternoon and good evening, everyone on the online. I'm Dejan Krajcar. I lead clinical innovation in Sandoz, which is a team focusing on optimizing clinical trials, and we spent a lot of midnight oil optimizing bioequivalent trials on oncology patients. As we heard today, and as all of you know, there's a unique set of challenges that are brought with this type of studies. And today I'm going to take you through some cases on how we address these challenges through adaptive designs, through replicate designs, and use of population PK modeling. I'm going to summarize some challenges quickly that I'm going to be mentioning today. So this touches uncertainties regarding the inter-subject variability that is usually tackled by pilot studies in healthy bee settings. However, it's not so easy to do in patient bee setting. There's a limitation in terms of volume of blood you can take from these already vulnerable oncology patients. There's a challenge recruitment and high dropout rate, which makes this data that we collect extremely valuable. I call like diamonds. And these studies are multicentric, which brings challenges in terms of protocol deviations. These studies are more prone to protocol deviations and harder to standardize. There are statistical implications, like you need to include the center in statistical model. First example I'll show you today, it's olaparib. It's a cytotoxic drug, and as we've seen in the previous presentation, it calls for bioequivalence trial in oncology patients and measurement of PK in steady state. For this drug, there was not possibility to run a pilot study due to the ethical cost and timeline perspectives. So the question was how to address the uncertainty regarding the inter-subject variability. We could plan standards 2x2 crossover design with a very conservative variability of 43%, but that would lead to high sample size and potential recruitment challenges. So we decided to go with an adaptive potency design, which is well known, and we use less conservative variability to plan it for the first stage, and then we would have an opportunity to increase sample size in the second stage if we are surprised with high variability. Luckily, we weren't. We passed the study in the first stage with enrollment of 70 patients, and overall, this is a 35% less patients we have included because we used this approach instead of a conservative standard approach, and it was 1 year faster. So don't forget, if you plan these studies, if you will be too optimistic in the first stage when planning a two-stage design, you might end up in a second stage, and then overall your total sample size due to the alpha correction might be increased. It's something to consider. So do your simulations also, because you need to include center and stage also in the second stage in your statistical model, and this has implication on your power. All of the examples I'll show next are masked due to confidentiality. So this is a product that again requires patient-based study on oncology patients, measurement of PK in steady state. So there was uncertainty again regarding variability, and we had options as before to plan a standard bioequivalent trial in 2x2 design with conservative variability of 35%. There was then option of adaptive design we had on the table, but then we thought also about the replicate design, which was already shown on one of the first slides by the presenter. So this is a great approach doing this replicate design, because you're not only tackling high variability that you might observe in the study as a surprise, and you can widen the confidence limits. But even if the variability is lower, you're actually also tackling the recruitment challenges that you have, because the sample size will always be half of what you would see in a standard 2x2 crossover setting. So sample size is reduction by 50%, but you're prolonging your study just by 2 days. So you're just adding one more replicate, which a trade-off is, of course, more samples taken from the patients. So we tackle that by doing simulations with PK model. I'll show example on the next slide, which is a similar one. And here we reduced volume by 25%. There are other benefits of this design, which is, you have better estimates of you have more accurate PK estimates, and design is more resilient towards statistical outliers or protocol deviation, which is also, it's not trivial in this kind of expensive and big studies. In the next slides, I'll try to highlight the increased importance of population PK modeling in planning and executing big trials on oncology patients. Since we are dealing with very valuable data, it's important to understand how occurrences in these long trials, such as missed or delayed doses, missed or delayed sampling points, or non-adherence to, for example, meal restrictions, would impact your study. It's important to understand which of these occurrences are not introducing bias to our study, because this would allow you to retain the patients that were affected, but are actually, it would be an unnecessary exclusion. So the data-driven decisions to make these decisions can be driven by population PK models because they are useful to simulate trials in different designs, different settings, different scenarios. So example I have on the next page is how we utilize population PK model for optimizing sampling points. This is a simulation of 100 studies in the similar full replicate setting that you've seen in the previous slides. Here you see a mean PK profile where we went from a full, we call it full schedule with 22 samples. We then progressively decrease the sample size, the sampling points first in the elimination phase, down to 15 sampling points, where you can see essentially no impact on geometric mean Cmax and AOC, or impact on T/R ratio or variability. But then we also went further and reduced it to 9 and 6 sampling points, where you can see Cmax and AOC being underestimated for about up to 9% Cmax and up to 3% for AOC. The impact on T/R ratio was negligible, and impact on variability just 1% increase in the scenario of 6 samples. Below 6, you go into the sparse sampling area where you would need to, the NCA doesn't work anymore there, and then you would need model-based bioequivalence to do that. For this case, we selected 15 samples. The question also for the discussion maybe in the afternoon is whether, you think maybe we could go lower because, we underestimated here Cmax and AUC, but we are not affecting here the conclusions of T/R ratio and variability, which is relevant for bioequivalence. Needless to say, this is an API agnostic conclusion. So you have to do simulations for your own API and explore wider range of T/R ratios, not just 100%. This is an example of using population PK model for missed or late doses in a similar replicated design. Here, in this case, we simulated missing doses in period 1 of this replicate design before we took the samples for steady state assessment and period 1 and period 2 PK assessment. In the real world scenarios, in long studies, this is likely to happen. People miss the dose, people take the dose, patients take the dose later than scheduled. Here, for the purpose of making a point, I'm showing results of extreme case simulation where all of the subjects would miss the dose in period 1 for RLD on day 2, or would have a late dose on day 2. And compared to the normal schedule, still, even due to this missed dose and even due to this extreme scenario, all of the subjects were attained steady state and had no impact on geomean PK parameters in period 1 and period 2, and there was no impact on T/R assessment. So it's a thing to consider when we are thinking about how resilient our design is towards this kind of events. And when we are defining protocol criteria, of course, we have to, in terms of administration, follow the label. But when you come to the gray area, which is not described there, so this is a great tool to use. Again, it's some API agnostic. You have to run for your own API. If, for example, API has a larger accumulation, then in this case, it likely also might have an impact if the dose on the day 2 is missed. So bringing some discussion points for the afternoon as well, maybe. I've shown you some examples of how we can use the population PK models to support decisions during the study. The question is, would such models that are just qualified on the literature data be okay to support this decision-making on the protocol deviations during the study before we see study results, and in what extent? The second question is, I've seen also there might be a similar talk in the afternoon about this. So interesting to see that about whether such models would be also useful for justifying exclusion of outliers, statistical outliers, if they happen in the study, if this is, of course, pre-specified in the protocol. I would like to I will end my talk here and thanks all of the colleagues and Sandos who worked on these studies, who helped with the design, the modeling, Rebecca, Kaya, Mojca, Petra, Primož, Mojca, Maja, Sasho, Igor, Lima, and Simon, and all others that I haven't named. And they were extremely important part of the development team. And I really mean this. I really thanks also Sandos for giving opportunity to develop these complex products that is not easy to make, and that we continue to increase the accessibility for patients. And in this case, I also would like to thank FDA for all the interactions that we can have with them on the other side to bring products to the patients. So big thanks to CRCGs for invitation to speak today. Of course, everything I shared today are my views and not represent necessarily views of Sandos. It's not QA time yet, but thanks for your attention. I leave you with a nice picture of spring in Slovenia. I've taken it a few weeks ago just to remind us, since we're talking about optimal designs, what kind of optimal design Mother Nature can do. So, and it's also an invitation if you come to Slovenia to check this. Thank you.
I guess I'll have to introduce myself. So I'm Yuqing. I'm lead pharmacologist from Division of Quantitative Method and Modeling from ORS OGD FDA. So my talk, following the Jen's talk, I will be talking from FDA perspective, how quantitative medicine approaches can help to conduct more efficient patient PKBE study. So as the previous speaker mentioned, sometimes the PKBE study needs to be conducted in patients when safety consideration preclude the use of healthy subjects. In such situations, BE study should enroll patients for whom the drug is intended to treat without disrupting patients' ongoing treatment. And in product-specific guidances, PSGs, for oral products that recommend in vivo BE study in patients, majority are for oncology drugs. This is a survey conducted by our colleagues that were presented at ASCPT 2024. So some challenges in patient PKB study, I think some other speakers already mentioned, including recruiting difficulty, high dropout, high variabilities, large sample size. And for steady state study, it will have long study duration and also difficulties in collecting intensive blood samples in patients. So in my talk today, I will talk a few cases where quantitative medicine approaches are used to support more efficient patient PKBE study. So my first case, I will discuss a recent PSG revision FDA did to exanomeb oral capsule, where we use the modeling to recommend alternative designs, replacing the steady state study. And then secondly, I will also share from FDA perspective, the alternative design. I think Dejan mentioned the TTRR design, the repeated design, as an alternative to the conventional fully replicate design for highly variable drugs with shorter duration in patient steady state BE study. And then I will share FDA's collaborators results regarding using model integrated evidence, MIE, to support alternative study designs and sampling schemes. And then I'll briefly touch upon adaptive designs in BE studies. So before I go to the detail, just briefly mention about steady state study. As many patients, they are receiving multiple doses. So in those scenarios, generally people are conducting steady state study. So that means it will need sufficient treatment doses to reach the steady state, as well as after switching. So it will bring the challenges such as the long study duration to achieve steady state, and what is the method to verify achievement of steady state. So here I want to share my first example. This is a recent PSU revision for exanomeb oral capsule. In the draft PSG that was originally recommended in February 2018, we recommend a steady state study in the target patient because this drug is taken as multiple dose. But then when we further look at this drug, based on the drug dosing cycle, so this drug is administered as a 28-day cycle, where the drug is given on day 1, 8, and 15 of the 28-day cycle. So as you can see, the typical PK profile here, this unequal dosing intervals will lead to difficulties to performing or confirming a steady state study. So in the recent PSU revision that was published in December 2025, we recommended two options. The first option is the BCS-based biowaiver option, where my colleague Gong later on will discuss how FDA's vision in BCS-based biowaiver. And the second option is the in vivo BE study with PK endpoints. So here, I'll go to a little bit detail here. So instead of recommending the steady state study, FDA conducted PK modeling to assess which dosing cycle and which dose would be sufficient to conduct the BE study. So here we are recommending the study to be conducted in a 4-cycle crossover design where patient receive 2 consecutive cycles of one treatment, followed by 2 consecutive cycles of the alternate treatment, which in the figure, the PK profile here. So the patient in cycle 1 and cycle 2, they will receive the test product, and in cycle 3 and cycle 4, they will receive the reference product. And in the crossover design, they switch the sequence. And then we recommend to collect the blood sample on the 3rd dose, which is day 15 of the 2nd treatment cycle for statistical analysis. So this recommendation is based on our internal population PK modeling, where we assess the different study design and to inform this specific cycle and dose is adequate to assess formulation difference. So my next example, I want to share our internal research regarding this repeated design, as Dejan mentioned, where it's instead of the polyreplicate design, the TR-TR sequence, this repeated design follows 2 repeated dose, TTRR and RR-TT sequence. So as shown in the figure here, it compared to the conventional polyreplicate design, the TR-TR design, as the treatment only switched once, so it shortened the study length compared to this. And we are taking 2 consecutive PK measurements for both the test and the reference products under steady state conditions. Since it has 2 repeated doses, it can also be used for reference scale average BE if high intra-subject variabilities are observed. Here I'm showing one figure from our internal simulation based on population PK modeling of example drugs. So the red line here in the figure is showing the study power for 2-way crossover study under different simulated TR ratio. And then the green and the blue line shows the conventional polyreplicate design, the TR-TR design, and the repeated design, the TTRR and RR-TT design. So our simulation showed that the TTRR and RR-TT design demonstrated similar BE passing rate as the conventional polyreplicate, the TR-TR design. And both this TTRR and the conventional TR-TR design showed much higher study power compared to the 2-way crossover design. And our simulation also showed the reduced estimates of intra-subject variability observed in the TTRR design when we compare to the conventional polyreplicate design. This is primarily due to the correlation of PK parameters between 2 consecutive sampling periods. So my first 2 examples, I'm showing how we use the modeling to inform a more efficient study design. Then my next example, I'm trying to do one step further to show you how model integrated evidence can be used as part of the assessment. So this is MIE-BE framework with population PK modeling that was proposed by our collaborator from Uppsala University. And I'm taking this figure from their publication. So in this MIE-BE framework, they proposed 4 steps, including model fitting, uncertainty estimation from the modeling, and simulation. And then the BE determination, it's made based on the simulated mean ratio of Cmax and AUC, and the 90% confidence interval is taken based on the distribution of the ratio mean. And I will use this example to show you how they use this MIE to support different sampling design. So this is a figure from their recent publication. So the authors demonstrated that using this proposed MIE approach for BE assessment, it exhibits higher power than the non-compartmental analysis method, especially when the data becomes sparser or more variable. In this figure here, the NCA-based method is shown as the pink bar, and then the other 3 bars shows the MIE method with different uncertainty estimation methods. You can show those MIE-based methods are always having higher study power when it compares to the NCA method, especially when it has higher variability and when the sampling is sparser. So the theory behind it is by leveraging population PK modeling, information is shared across subjects, so it enabled the method to handle sparser data more effectively. And again, in this MIE approach, it involves fitting the model into the PKBE data, then using this model parameter estimates and their associated uncertainty to derive the ratios of model-based PK matrix, AUC and Cmax, and their confidence interval, which are used to assess BE. The same group, they also use this MIE approach to support alternative crossover design with incomplete washout. So their results showed that MIE together with reference scale B analysis are flexible enough for analyzing data for novel designs with an incomplete washout, which they showed adequate study power and type 1 error control. And simulation showed that the incomplete washout design reduced the overall study duration for about 70%. I think this is especially very helpful for patient study, as in patient study, you cannot disturb patients' ongoing treatment, so it doesn't allow to do washout. However, in this design, it adjusts the carryover in crossover study when the washout is not visible, basically using the modeling to remove carryover. So it allows more flexibility in the study design instead of doing the conventional steady state study. Last but not the least, I will briefly mention about adaptive design. As the previous speaker mentioned, adaptive design sometimes quite useful as it allows to prospectively plan modification for one or more aspects. Adaptive design can be group sequential design or other design with one or more adaptive features. So it provides the possibility of stopping the study earlier or adjusting the sample size during the conduct of the study, like the pop-in method showed previously. And here I want to share a paper we previously published where we collect the adaptive design that was submitted to OGD and the application of those adaptive design in generic drug development. And I also want to briefly share there were more features can be included in adaptive design. In the recent publication from one of our collaborators, they showed the model-based approach can also be combined with group sequential and adaptive design. And I'm providing a link to their publication here. So this is my last slides. I'll wrap up my presentation here. So today I showed a few cases where quantitative approaches can help to develop more efficient and feasible patient-based studies. So at OGD, we see a clear demand that increased use of quantitative approaches to support alternative designs in pre-ANDA interactions and ANDA submissions. So we encourage the generic applicants to develop alternative approaches and engage with the agency early during the development. And it can be communicated through control correspondences, pre-ANDA meetings, and specific questions regarding MIE approaches can be submitted to MIE pilot program. And I'd like to acknowledge my colleagues, Mark, Paige, and my management, Andrew, Lucy, and my leadership, Rod and Lei. Thank you. And our next speaker will be Dr. Lalit Kumar and from Sun Pharmaceutics, and who will be present remotely.
Hi everyone. So thanks to my predecessors, the speakers, to enlightening the group on the current insights on the new BAB trials and recommendations. My topic is a bit different from the earlier speakers, and it is more related to IVFBC. My topic heading is leveraging early dissolution risk assessment and PPPM to predict its clinical impact. Here is a disclaimer. The views expressed and opinions expressed in the presentation are solely those of the presenter and necessary, not the view of the Sun Pharmaceuticals. Moving to the next slide. So this is a summary slide. So this highlights basically challenges faced by generic formulation development and risk mitigation approach. Usually, generic formulation development, which is an example in current case studies as well, shows a non-similar dissolution profile as compared to RLD. This may be due to the difference in formulation technology. The dissolution difference is more pronounced at lower strength. This may be due to the reduced drug load. As for a lower strength, drug load is decreased, so solubility of zinc condition is increased, making the dissolution matching more difficult, and hence profile matching or matching F2 across all strength required significant formulation changes and may go beyond the SUPEC level as well. So risk mitigation approach is the model-informed drug development with an exploratory design space offered a structured mitigation pathway. This also enables the decision making without excessive formulation reengineering. An exploratory clinical study was conducted in a current CES to define the bioequivalence P design space, and further PBBM modeling was used to predict the virtual bioequivalence for the lower strength and hence support the lower strength non-similar dissolution, and this can also act as a risk-based strategy supporting the non-similar formulation development, whether it's a lower strength or maybe a specification setting. So moving to the contents. So my slides contents include a regulatory framework, formulation development challenges, alternative approach, and a final case study, which is a case study X. This slide indicates the background and regulatory framework which a formulation colleagues has to refer by developing a generic formulation. First is the M13A guidance, which is a criteria for conducting a B study, PK endpoint for orally administered IR solid dosage forms. As per this guidance, normally the study design for any study is a single dose 2 treatment 2 period crossover study. Also, the M13B guidance describes the 5 considerations for additional strength not to be investigated in BE studies. This includes the acceptable in vitro dissolution testing on lower strength in comparison to higher strength. Also, proportionality similar of all strength, making the lower strength a bit challenging as compared to higher strength, specifically for a BCS class 2 or class 4 molecules. And specification settings are as per reflection paper. And specification settings are mostly based on the clinical and biopharmaceutic specs. That is, if dissolution of a biobatch is larger than or equal to 90-95% in 15 minutes, then specification may be set to 85Q in 15 minutes. And again, the trace test guidance is also there, which indicates that the RLD should be similar in trace test to test product should be similar in guidance. If any of the approach has not to be followed, then IBABC or modeling approach to be required to support the bioequivalence as per the relevant regulatory agencies. So again, we discussed the formulation challenges that meeting a generic formula with respect to RLD on a higher strength. If it is not met, then the clinical bioequivalence study is required, which is the part of B guidance, but bi-waiver can't be achieved if the lower strength in dissolution is not similar to higher strength, or the stringent dissolution specification, as the guidance says that the dissolution specification to be stringent. So in this case, if the requirement is there is some relaxed specification criteria or lower strength is not matching in dissolution to higher strength, then additional clinical studies is required. These multiple studies in oncology may be challenges due to specific population involved and safety. So In next slide, we will talk about alternative approach. An alternative approach is to the initial formulation development to cover the product's design space, and PPPM, PPPK modeling, development and validation, and then apply this develop model to compare the multiple formulation to set specification range, B strength for higher strength and lower strength, or BB strength for lower strength, generic versus RLD. So we'll discuss that with an example. So products X, this products X is a oncology product, BCS class 2 oncology product. The solubility is pH dependent solubility, soluble in acidic pH, but solubility decreases after that. Absorption time Tmax is around 1 to 4 hours. Bioavailability is 10%. With food, bioavailability increases. Plasma distribution, apparent volume of distribution is greater than 5,000 liters. Elimination half-life 30 to 50 hours. An adverse event is that normally a higher strength is given, but if a higher strength shows adverse event, then shift to lower strength. So basically points to highlight is that this is a BCS class 2 class 4 type of molecule which shows a higher half-life. So it is also indicated that bioavailability is limited in fasting condition. That's why under fed condition bioavailability is improved. Apparent volume of high apparent volume of distribution. So this is a class 2 class 4 molecule with high volume of distribution and high half-life. So coming to the drug release characteristics, and the case I want to show here is we will focus first on the left hand side that indicates the dissolution profile of test and RLD. So in this case, if we see a dissolution profile that is shown in blue and orange, then higher strength of RLD differs from lower strength, and lower strength is lower in dissolution profile. While if we see the two prototypes, or one from the higher strength and one from the lower strength, then in case of a test product, reverse trend is observed, that is dissolution profile for the lower strength is faster. So this is a case study where a comparative release between test and RLD may differ due to difference in APMO or difference in excipient. So faster release is observed in a lower strength where the complete release observed in 15 minutes, and we can see a difference in lower strength test and RLD. So moving to how the design space was created. So two test prototype of higher strength, if we can see that differs in a 20% in dissolution profile. One higher prototype 2, which is a faster in dissolution profile, approximately 80 to 85% release in 15 minutes, and another slower prototype 2 RLD was subject to B dosing. So on the right hand side there is a plasma concentration curve. So if we can see that the both prototypes differs to RLD with in less than 5% difference in PK parameters, indicating that two profiles, although there is a difference in dissolution data, but two profiles are similar. So further, this type of design space are explored further to study the impact of dissolution time point, address lower strength similarity, and specification designed to address the edge of specification failure. So model structure and application strategies, model development and validation through literature and announced data. Population simulation of 50 subjects with gastrophysiology as a crossover design, and PK parameter Cmax and AUC for each strategies was run as a two-set variable, and then virtual bioequivalence was determined. Application was carried out is lower strength versus higher strength within the test product, lower strength test versus lower strength RLD, and specification puzzle setting. So first application is the VB predictions for lower strength versus higher strength. So we can in the earlier graph we have seen that the lower strength is faster, but if we have predicted the VB results, then these two sets of virtual bioequivalence indicated that although the dissolution is non-similar, but expected to meet the bioequivalence result. So faster profiles are not translated into higher T-by ratios in the actual BE study as well as the in silico calculation also. This may be due to the low permeability of the molecule, so permeability limited, so dissolution is not translating into the actual BE data. Again, the test lower strength versus RLD lower strength, we have seen that drastic difference between test and RLD dissolution profile. Test dissolution profile was faster while RLD dissolution profile was slower. Again, the VB was 1 and the VBs indicated the similar results. Then moving to the specification proposal. So here first we'll discuss the graph. So here the graph indicates the orange, blue and gray line that we have dosed in a BE study, and the BE was a bioequivalent formulation for the faster and slower prototype. So if we consider the TP2 fast release formulation as a final prototype, then as per the guidance, the specification is ATQ in 15 minutes, or if a stringent reviewer may go for a stringent, then 85Q in 20 minutes. This is much stringent specification. So again, a side batch concept is there in concept paper. So as per that, if we consider the lower dissolution set, that is based on the TP1 formulation, then we can say that 85Q in 30 minutes. But how if we want, if it needs to be determined through a design space or a modeling type? So the predictions for 3 cases have been considered. One is ATQ in 30 minutes, ATQ in 45 minutes, and ATQ in 60 minutes. So this is the VB prediction. We can see that for proposal 1 and proposal 2, it led to a similar B result and passing B criteria. However, considering ATQ in 60 minutes, is that where a B failure was observed. So specification limit of ATQ in 45 minutes considered for considering a clinically relevant dissolution specification. And in the right, there is a design spaces there. We that can certainly say that anything above ATQ in 45 minutes should meet the B criteria. So in this way, design space has been generated. So moving to the key takeaways with available exploratory studies and science-based justification. So here lower strength is similar to higher strength in test formulations. If we compare it with lower strength to RLD, then also it is similar, and then there is a option of specification setting based on the side batch rather than the target batch. So here just a points to ponder. So in this case, since the dissolution for 2 prototypes are greater than 20% and still the products are bioequivalent, so drug pharmacokinetic properties are also critical factor in BE. So molecule with high volume of distribution, high half-life are less likely to show bioequivalence challenges. So it is not always the pH solubility, that is BCS class 2 and 4 may be criticals. So Bioweaver criteria for lower strength that determines the dissolution similarity that consider the formulation factors should also keep in mind the drug pharmacokinetic properties and should be relaxed based on the pharmacokinetic properties. So this is from my end. Thanks all, and thanks to my superiors for presenting this case study. Thanks. Thank you, everyone.
Thank you, Dr. Khurana. So I apologize, I could not give a chance to introduce which speaker because of time tangent, but I think our session semi on time, so I could have a chance to introduce the next speaker. Next speaker, Dr. Emilija Fredro-Kumbaradzi. Dr. Fredro-Kumbaradzi is the Director of Biopharmaceutics and Statistics Apotex. Her presentation title is Advancing BCS-based Bioweaver for Oncology Product Through Risk Assessment.
Good morning and good afternoon. It is my pleasure today to speak in this workshop on the topic of advancing BCS-based biowaivers for oncology products through risk assessment. My disclaimer that the views expressed here are those of the author and do not necessarily reflect a position of my organization. This is the outline of my presentation. I'll briefly start with generic industry challenges, move into impact of these challenges on generic oncology development. What are the strategies that industry has to address the challenges? Then I will focus on BCS biowaiver opportunities for cases which do not fully meet ICHM-9 requirements. And I will present a case study where biopharmaceutics risk assessment is utilized to enable BCS-based approach when ICHM-9 criteria are not fully met. I will not go over generic industry challenges because we heard all what patient study bring to generic industries. So I'll skip it in the interest of time. What would be the impact of all these challenges around patient studies, costly, lengthy, complex? It's an increased development risk and uncertainty. Of course, economic feasibility is reduced, which may impact smaller manufacturers, delayed market entry for generics, and of course, potential limited competition will impact patient access to medicine and affordability. What would be industry strategies to address these challenges? Of course, industry would focus on finding alternate regulatory pathways to minimize patient studies, increase reliance, for instance, on BCS biowaivers would be one option, biopharmaceutics risk-based approaches, thorough in vitro characterization, modeling and simulation to support the bioequivalence argument. In order to get regulatory feedback on potential alternative regulatory pathway, industry often engages in correspondence with agencies to get some clarity. Let's talk about BCS-based biowaiver opportunities. We all know there is ICHM-9 guidance, which clearly defines biowaiver requirements. If they are met, path is very clear. Those requirements are typically made of three parts: solubility information, which can be supported with in-house data, permeability or absorption-related information, which generic industry often looks for in literature. And here is to be noted that we face challenges with accepting mass balance data from literature by the regulators. The third part are dissolution data, again, that can be supported with in-house experiments. But what would happen if criteria from ICHM-9 are not fully met? For instance, if there are issues with stability during solubility study, more than 10% degradation, if there is a conversion of the product to drug pre-systemically, when we need to know how much is converted and what would be the impact on bioavailability, degradation in GI environment with as simple process as hydrolysis, or for BCS3 biowaiver, departure from Q1, Q2 composition, can we assess the differences and their potential impact on bioperformance? Also, sometimes dissolution criteria not being met by ICHM-9 could be a challenge to justify. So, what would be the BCS-based biowaiver justification options? I would like to present one case study, which is a BCS3 drug, a PhD recommend study in patients. Considering its BCS3, we wanted first to explore pathway for BCS biowaiver. However, there is a challenge that we faced, and that is that although it's highly soluble, it's unstable in acidic environment with significant degradation below more than 10%. So we need to assess the impact of degradation on bioavailability. To do so, we need to determine solubility and stability across the physiological pH range, determine the degradation rate at acidic pH range in order to utilize biopharmaceutics risk assessment, where we combine knowledge on physiological perspective on this degradation, effect of fasting stomach pH on degradation, and we utilize PBPK modeling to virtually assess the impact of this degradation on the bioequivalence between the test and reference product. Drug is highly soluble, as I mentioned, and degrades more than 10%. On the right-hand side, you see a graph that shows stability at different pH. The steepest curve is obviously for lowest pH. ICHM9 criteria are not met because of the degradation. However, degradation rates for test and reference are similar. We need to conduct biopharmaceutics risk assessment to assess what would be the impact of this acid instability on bioavailability and bioequivalence to the reference product. This is the dissolution profile of the test and reference. It's very rapid across the physiological pH range for both products. Obviously, dissolution is not a rate-limiting factor for absorption, as it is expected for highly soluble drugs. In order to assess the effect of the degradation under acidic conditions on bioperformance, we developed a PBPK, starting with disposition model when we utilize in-house data and literature on API properties and available literature information for IV administration. As shown on the graph, simulated and observed PK profiles for IV administration were similar. Oral absorption model was developed when solubility and dissolution profile for the reference, which is, as I mentioned, very rapid, along with the degradation rate were input in the model to simulate a PK profile of the reference under fasting conditions. As shown on the graph, simulated and observed PK profile, which we've taken from literature, are similar. Model validation was then performed utilizing PK data for reference product when co-administered with PPI under fasting condition found in literature. The graph shows simulated PK profile, which is compared to the observed PK parameters, Cmax, AUCT, AUC-INF, available in literature and found to be similar. We have also conducted parameter sensitivity analysis for stomach pH and dissolution. Of course, dissolution or Z factor does not impact much, and stomach pH is the one that can impact Cmax and AUC both. In order to assess the impact of degradation on bioequivalence of the test and reference, we use this developed and validated model, and the plot shows plasma profile for a reference observed found from literature, reference simulated, and test simulated. But bioequivalence between the test and reference was demonstrated virtually. In conclusion, BCS-based biowaivers can be alternative B approach for oncology product. And if ICHM9 criteria are not fully met, we can consider biopharmaceutics risk assessment to see if there is a possibility scientifically sound for a biowaiver pathway. Experiments along with modeling may open the scientifically sound alternative pathways. The case study clearly showed that BRA supported with PBPK model demonstrated there is no impact of degradation on bioequivalence. PSG recommendations were updated subsequently with BCS3 biowaiver option. At the end, I would like to acknowledge support from my Apotex colleagues, Naveen Vaya, Pradeep Sangvi, Kiran Krishnan, Fei Han, and Sumon Chakraborty. Thank you very much for your attention, and allow me to introduce the next speaker, which is Dr. Li Gong, lead pharmacologist in OGD at FDA, who will continue talking on BCS-based biowaivers for oral oncology products. What are the general considerations, challenges, and case studies? Thank you very much.
Thank you, Emilija. So next speaker, Dr. Li Gong, please.
Good morning, everyone. I'm covering my dear colleagues, Dr. Yi-Hsien, but because he did not make this session of the workshop due to personal reason. So the title of my small talk is BCS based on biowaiver for oncology, oral oncology product, general considerations, challenges, and case studies. Okay, this is my disclaimer. This is my talk outline. I'm going to talk about challenges associated with in vivo PE studies for generic oral drug oncology product, and then talk about a little bit principles and general considerations of BCS-based biowaiver, and then case studies and summary. As we know, challenges is always with in vivo studies for oral oncology products. Historically, the in vivo PK studies talk about healthy volunteers versus patients have been recommended to establish PE for generic oral oncology products. The challenges including conducting studies, B studies in healthy volunteers, health subjects may not always be feasible, and concerns sometimes for cytotoxic drug product, and you have to required for submitting the bio INDs, and also difficulties in patient enrollment depend on the cancer types, stages, and treatment history, and those regimens, etc. Also, high cost, high variability requires large sample numbers. Disease, like disease progression, and committed medications, as well as the study design, usually for parallel study design. And lastly, and not least, high cost. We are talking about steady state study design and intensive safety monitoring, and along with supportive cares. So all these are a challenge for conducting in vivo BE studies for oral oncology products. So this brings us to think it is the BCS-based waiver as an alternative approach. So in this opening remarks, Rob mentioned the quantitative biopharmaceutics. And here I just start off a basic for four equivalent squares. So that is a scientific approach based on aqueous solubility and intestinal permeability characteristics of the drug substance. A drug substance can be categorized into one of the four BCS classes. And class 1 and class 3 with high solubility, with one being the high permeability, and class 3 with high solubility and a low permeability. For BCS class 2 can be categorized for high permeability and low solubility. For BCS class 4 drugs is low solubility and low permeability. And this is how high ability defined and the permeability, high permeability defined. Now, talking about eligibility of BCS-based biowaiver for drug substance requirements, solubility, and for permeability. For BCS class 1, high solubility and high permeability. For BCS class 3, high solubility and low permeability. And for in vitro dissolution, a requirement is rapid, that is to say, greater than 85% in 30 minutes for BCS class 1. For BCS class 3, and the in vitro dissolution is defined as very rapid, greater than 85% in 15 minutes. For drug product requirement for the formulation, for BCS class 1 is critical excipients required to be quantitatively, that is Q1 the same, and quantitatively similar. For BCS class 3, the formulation similarity requires for all excipients except for in film coating and capsule shells. For Q1 the same and Q2 similar. So these applicable to the immediate release solid oral products or suspensions designed to deliver the drug product to systemic circulation. The exclusions include the modified release, the drug products, narrow therapeutic index drugs, and oral cavity absorption drug products. Now let's move on to case studies. Case study number 1 is a cladribine tablets, which has acid instability issues. Actually, this is a neurology oral drug product instead of oncology, but the same principle applies. The RD is Mavenclad tablets, 10 milligram under NDA 022561, approved on March 29, 2019. So the indication is a relapsing form of multiple sclerosis. Previously, our PSG recommending the one single dose, 2-way crossover in vivo B studies in patients with relapsing forms of multiple sclerosis who plan to receive the first or second treatment course. The a cladribine substance undergoes degradation increased with the due to hydrolysis to the drug at acidic conditions. And also cladribine formulation developed using complexation of cladribine with 2-hydroxypropyl beta cyclodextrin, HP beta CD, to form a complex hydrobine, as you can see here. Now let's review what the ICH-M guidance for BCS3 class waiver by waiver for formulation requires excipient quantitatively same and qualitatively and quantitatively that's Q1 same and Q2 similar as RD. Also for solubility need high solubility with less than 10% degradation. And also for dissolution, it requires very rapid dissolution greater than 80% within 15 minutes. In our cladribine case, we found the Q1, Q2 formulation similar is acceptable. The solubility data shows greater than 10% degradation at acidic condition. And also the dissolution less than 80% profiles in acidic conditions. So both solubility issue and the dissolution issues related to API stability at acidic condition. So which brings our questions whether still eligible for cladribine for BCS3 by waiver. So after review all the data submitted, and we found the application contains suitably validate stability indicating analytical method. That is to say, they showed the API's solubility and also the detection of the degradants by demonstrating with mass balance studies. And also both API and API complex were evaluated separately, and cladribine separates from cyclodextrin before absorption. And also the data shows 2-hour time frame suitable for solubility at the acidic conditions. So let's say average gas transient time and the cladribine Tmax range. And for by relevant data in fasted state stimulate gastric fluid FASGIF, which is under a pH of 1.6 and more basic compared to very acidic conditions. So the solubility studies, the API and API complex is considered much higher in the than the threshold to be considered the highly soluble, which is 0.08 milligram per ml as API, so up to 4 hours. For dissolution, very rapid dissolution, which is safe to say greater than 85% within 15 minutes, both for test and reference at the FASGID media. So another thing from the NDA clinical data, the co-administration of pentoperazole, the proton pump indicator inhibitors PPI did not have impact on the PK profile of cladribine. That is to say, no effect on rate and extent of absorption. So another evidence is by using supportive PBPK modeling, they evaluate the impact of drug degradation on in vivo performance. That is to say, they incorporated the physical-chemical property data, gastric degradation kinetics, and also bio-relevant dissolution profile into PBPK model. So the model was then validated against clinical PK data. So it is concluded that simulation using the optimized model showed that degradation of cladribine at acidic conditions is not expected to impact the B under both fasting and fed conditions. So based on above, the BCS class 3 waiver based waiver for cladribine is feasible, as supported by the above totality of evidence. So let's move on to case number 2, designated as a drug X, which also has acidic instability issue. This is the oral oncology drug product and a potential BCS class 1 drug. And after evaluating submitted data, we found the formulation, dissolution, and permeability data are all acceptable. And also higher solubility at middle to high pH conditions. Only challenge is the API underwent degradation greater than 10% within 60 minutes only at a low pH. So on the basis of scientific justification, we checked the RD label. It says the drug X should be taken with food only. So actually, under the fed conditions, the gastric pH is in the middle of pH range. So therefore, the acidic acid instability may not be physiologically relevant. So drug X can be categorized BCS class 1 drug. So on the broader principle, BCS by waiver may be feasible if evidence demonstrates that in vivo degradation, in vitro degradation does not significantly impact on the in vivo performance. So the drug X is eligible for BCS class 1 waiver. Move on to case number 3. It is designated as a drug Y, a prodrug. It is also an oral oncology drug product. Drug Y is a potential BCS class 3 drug. Per current PSG, it recommends a steady state in vivo B studies in patients. So according to RD label, drug Y is a prodrug that is rapidly hydrolyzed under physiological conditions to its biologically active form, that is free base form. So the drug Y shows pH, also drug Y shows pH dependent solubility, meaning increased solubility as pH value increased. So after evaluating all data submitted, we found acceptable formulation and the dissolution data. The only challenge is decrease the solubility over time at higher pH, for example, a pH 4.5 and pH 6.8 up to 24 hours. But we found that the stability data is acceptable. So question is drug Y eligible for BCS class 3 waiver? Let's address the first challenges. That is the drug absorbed as a prodrug or active form, that is free base form. So according to ICH M9 guidance, it says prodrugs may be considered for a BCS biowaivers when absorbed as prodrug, and unless the metabolite is formed immediately following intake and applied to absorption. So actually, drug Y as prodrug rapidly hydrolyzed to its free base under physiological conditions without enzyme needed. The conversion occurs immediately upon exposure to squares aqueous solution or plasma. The MMR spectroscopy data shows only the free base is present in solution, confirming the complete conversion under intestinal pH conditions. A free base actually is an analyte of interest for both in vivo PK and in vitro dissolution studies. So it is concluded that conversion to free base in physiological condition can be occurred immediately prior to intestinal absorption. So our second challenge is for this drug product is decreased solubility over time at a little bit, middle pH 4.5 to 6.8. Is it a concern? So basically after the review the data submitted, we found the equilibrium solubility study were conducted with an acceptable study method and saturated that. So basically, pH dependent solubility, there should be as pH increases and solubility increase. Why decreased the solubility occurred? It may contribute to the prodrug conversion to less soluble the free base after called a phenomenon known as the precipitation out. So under saturated condition. So drug concentration in solubility study, saturated condition being substantially higher than the concentration and the physiological condition, that is therapeutic dose. So overall, the solubility meets the high soluble criteria, much higher than the threshold to be considered highly soluble at all pH conditions. So it is then concluded that decreased solubility is not a concern in vivo condition. So drug Y is eligible for BCS class 3 waiver. To summarize, the BCS-based waiver can serve as an alternative approach to establish BE for generic oral oncology product, especially when the in vivo studies presented significant safety or feasibility challenges. Submissions utilizing BCS-based waiver approach should follow the recommendation recommended provided in the current M9 guidance. Any deviation from the current recommendation in the M9 guidance or PSG must be supported by proper scientific justifications. So finally, regulatory evaluation of a BCS by waiver request should be based on the totality of evidence. Therefore, the acceptance determination of the requests can be only made upon comprehensive review of all submitted data. With that, my acknowledgement to the OB and DB3 management for their constant support and encouragement. For our DB3 review team members for their dedication and hard work and present the case, moving some of the cases forward from submission to approval. My acknowledgement also goes to our colleagues in EQMM and at ORS for their management, for their assessment teams, offering the great collaboration, providing their expertise, the consultation, and as well as supportive expert in PBPK modeling and simulation. And lastly, we'd like to thanks for CEDAR's BCS committee members for their patience, for their support, and basically they cast their favorable votes for these three above cases. With that, I would like to turn over to presentation of next speaker, Dr. Arindom Powell. And Dr. Arindom Powell is a pharmacokineticist in EQMM, ORS, and OGD. So his topic of the presentation is PBPK modeling to support the alternative B approach for oral oncology drugs. Thank you for your attention.
Hello, everyone. I welcome you all again to this presentation, which is on PBPK modeling to support alternative B approaches for oral oncology drugs. This is the standard disclaimer that these are my views, not FDA's. Outline of this very short presentation is I'll just touch upon the introduction, provide some examples on the PBPK modeling in generic drug submissions, provide the case examples, and leading to conclusion. So when we talk about alternative approaches, the totality of the evidence for alternative B approaches may integrate biopharmaceutics and clinical pharmacology information, in vitro testing data, chemical manufacture controls, release mechanism understanding, relevant clinical data, as well as PBPK modeling and virtual simulations. As you know, PBPK modeling can integrate drug-specific properties, physiological parameters, formulation characteristics, and population variability. Therefore, it has a great potential to support the alternative B approaches as a mechanistic bridge. So all my previous presenters has talked about the challenges of oncology drug B assessments in detail. So I'll skip this part looking at the time. But I would like to mention that in the slide that PBPK modeling has emerged as a promising approach that can incorporate disease-specific physiological changes in cancer patients and also enable virtual B assessment by incorporating critical product quality attributes or bioavailability attributes, and thus provide a mechanistic understanding of drug absorption and disposition. Here are some examples of PBPK modeling in generic oncology drug submissions The left figure shows data up to 2026, and we have received about 22% of all submission in the category of oncology drugs. And of those 22%, modeling submissions, on the right table shows the uses they were for. For example, evaluate drug degradation impact on B assessment, justify a reduced sample size for the patient PKB study, evaluate alternative study design, specifically AABB or BAA, what we have seen in our previous presentations, virtual B demonstration under FET condition using PBPK, IVIVC models, justify dissolution difference across different strength using VBE and IVIVC. So I'm going to present a case from our internal research project on Olapari PBPK model. Purpose of that model was to evaluate PBPK model and virtual B simulations to support alternative B approaches for developing Olapari generic drug products, potentially address the inherent challenges of patient-based PKB studies while maintaining regulatory confidence in B conclusions. A little bit on Olapari. It's a putative possible BCS class 4 drug. The RLD Lynparza is an amorphous solid dispersion, so therefore it's categorized as a high-risk product that generally warrants the B study to be demonstrated under both fasting and fed conditions. The PSG of this product recommends one PKB study in patients for the products which are very similar in formulation design to the RLD. The PSG also mentioned that PBPK model can be used to justify any formulation difference that would not, a formulation difference would not impact BE along with other evidences such as comparative dissolution profiles and etc. Published literature on PBPK, Olapari PBPK model demonstrated that these models are basically used for predicting DDI, but not for B purposes. So in our research project for Olapari PBPK model development, we use this general workflow for the development, validation, application of the PBPK model. This is basically a middle out approach where we develop the the disposition model using literature data and also some of our in-house data. Then we use our in-house NDA and ANDA data to for the model development, validation, and parameter sensitivity analysis, as well as for virtual B simulations. I'll go straight ahead to our results. During the Olapari PBPK model development, we developed the model for both fasting and fed condition. And both single dose and steady state data were used for model development. And this figure, all this figure actually shows both fasting and fed condition. The last figure shows simulation under steady state condition. One thing to note that we are not showing, any observed data due to disclosure limitations. And we found that all these simulations showed great percent prediction error, all below 15%. For model validation, we, as I mentioned, we used NDA and ANDA data for different strength and both single dose and steady state simulation. And these models were validated sufficiently, demonstrating close prediction error for Olapari PK, where all the prediction errors were below 15% for the PK matrices. We also did sensitivity analysis using parameter sensitivity analysis and virtual simulations. Our PSS showed that Cmax was sensitive to PEF values at the lower end, but become insensitive when the PEF value is higher than 6 to the power 10 to the power minus 4 centimeter per second. It is understandable that as a BCS class 4 drug, that values is very high, considering it's a BCS class 4 limited permeability. The AUC was also found to be insensitive when the PEF value goes above 2 into 10 to the power minus 4 centimeter per second. Sensitivity analysis for dissolution was done using virtual BE simulations, where we used hypothetical dissolution profiles as an input. The reason being we did not have a specific study set where it was a failed BE. But the PK metrics, Cmax and AUCs, TR ratios were not sensitive to a large range of significantly changed dissolution profile as model input. That indicates that indicating the permeability may be a rate limiting factor for this drug, not the dissolution here, what is shown in the last two graphs, C and D. So we use VB simulations following the PSG recommendation that recommends to use steady state BE in patients. And using those VB simulations in multiple dose steady state crossover design, we found that the PBPK model predicted the BE between test and reference drug product, which was also in agreement with the observed data set under fasting condition in patients. We also did the VB simulation under fed conditions, where we also obtained similar result that the test and RLD was BE. So summary from these exercise, we can say this PBPK model demonstrated good predictive capability for both single and multiple or steady state dose steady state administration and virtual BE assessment. And the study demonstrated that PBPK-based virtual BE may provide a supporting evidence as a part of the totality of evidence for an alternative BE approach for such generic drug products addressing significant challenges in patient-based BE study. Overall, this study was actually a part of our research effort to explore the potential of PBPK model to support alternative BE approaches and to justify that difference in formulation design for BCS class 4 drugs may not or not impact BE in lieu of conducting challenging patient-based PKB studies. In conclusion, PBPK-based virtual BE may provide a supporting evidence for alternative BE approaches for generic oncology drug products, addressing significant challenges in patient-based BE study for this type of drug products. And for such applications, we recommend communicating early with FDA to discuss PBPK modeling strategies via a MI industry pilot program or pre-NDA meeting pathways. So I'd like to acknowledge my oral PBPK team, the DQMM management, and Yuqing. Also, like to thank Dr. Jihong and the ORS leaderships. And thank you all for your attention. We can take the question during the panel discussion. Thank you all. I'd hand over to the floor to Dr. Jihong.
Thank you, Dr. Parr. I would like to thank again all the speakers for great presentation. I know the bioavailability of bioequivalent division, some farmer. And Dr. Cheng? Okay, not here now. So we're going to start to address questions from audience in personal attendees. So any question from floor? Okay. Hello. Okay, we can hear now. Yeah.
So this is a question for the industry speakers. And so when you're planning your development plan for your study for an oncology drug, and you have the product specific guidance that says that you should be using only patients, but what makes every now and then you deviate from that? How do you justify that, especially if the context is that the concern could be for long-term malignancies, which you may not be seeing during the conduct of the study, but you would see afterwards. And what kind of stipulations do you have for following up those patients afterwards for those malignancies, should you decide to conduct it in healthy volunteers? Thank you.
So I wonder, Dr. Krajcar can
Yeah.
address that.
Thanks for the question. I would say that when you said from time to time we deviate from the recommendation, I wouldn't say that decision is taken lightly or without consultation with the agencies. So definitely when we see opportunity that, something needs to be reconsidered in terms of the selected or proposed population and the product specific guidance, we would reach out and really in details across different functions of assess, what is the probability that potentially we could propose study in healthy volunteers. So from that perspective, I would say that that would be a two-way discussions with the agency and not be just decision that we would take lightly and just do a study in health, even though there is a proposal to do it on patients.
Thank you, Dejan. But I may ask this question to Dr. Khuroo because he posed this type of question and when I collect some potential question from presenter. So Dr. Khuroo, would you like to make a comment on this question?
And we hope that's the case, that you do reach out to the FDA before you deviate from the PSG, especially when it comes to patient safety concerns. Go ahead.
Thank you, Michael.
So thank you. If I understand correctly, that question was on what basis or why should we deviate the guidelines when project-specific guidelines mentions that study has to be done on patients. See, it's not very simple, but it's not also very complex. Important thing here is safety. Now, if a drug is a cytotoxic drug, and even if a single dose will have cytotoxicity on healthy subjects, then there's no way of going to healthy subjects. We have to stick to patients. But if we have enough safety data, whether it is your local population or worldwide, any other paper is available which will confirm that drug is safe to be given in healthy subjects, then of course you can take decision accordingly. And you can also get in touch with the regulatory agency. I will just conclude this question by giving you one example, like sunitinib a couple of years back, I think 5 or 8 years back, in fact. OGD was mentioning that it has to be done in patients, but there were other guidelines, like Europe was saying it has to be done in healthy subjects. Then when we checked literature, there was enough literature available on healthy subjects, and then OGD also changed it to healthy subjects. So just to conclude, what I'm saying is that safety is the main important point here, which helps us in taking a decision whether to go for patients or healthy subjects. Thank you.
Thank you. Thank you for your, the comment. I think there might not be a significant gap, but probably case by case, agency made some decision, industry could make another decision. So that's the reason why we discussed this topic in this table. So I would like to jump to another type of question. Okay, any question from the floor? Okay.
I have a question for Dr. Khurana. He's, I think, online. Dr. Khurana, I enjoyed your presentation, including your example where you had a product with several strengths, and you mentioned lower strength waivers, and there was differences in dissolution between test and RLD, and that was maybe even amplified for lower strength. And then you end up using some what you call virtual bioequivalence examination. I guess my question is really a validation question. In that model component, the modeling component, what model validation challenge did you most face? It's kind of interesting about if model validation is a problem at all, and what was it, and how did you grapple with that?
Hi everyone. First of all, my voice is now much clearer than my presentation.
If my screen is too blurry, then it's some their Wi-Fi issues. And apology once again. So question is regarding the validation challenges. Yes, for a PBPK modeling where multiple parameters are involved, modeling is a challenges. But main point here is to address the part whether the in vitro dissolution is able to show the discrimination in vivo difference or not. Best example is to carry out a B study with much difference in dissolution data and then use that as a model validation. There may be other validation approaches as well, but my point here is to use that data as a validation. Again, reviewer can assess questions on the type of population taken, type of variable taken. That needs to be scientifically addressed. So is my point clear?
Yep, well, thank you very much. Thanks for participating.
Okay, I'm going to move okay. Think you have question.
Thank you. I have a question for you, too. You mentioned a design of AABB, BBAA instead of the traditional ABBA design to, sort of shortening the study time and reduce the subject number. Is this type of study, study design product specific or it's formulation specific? Can you generalize this idea to other type of oncology product? For example, I know in biosimilar, they do PK study on the solution injectables. Could this design also generalize to solution product, which is a different formulation? That's my question. Thank you.
I think to me, this design should be generalizable, because here, we are still collecting 2 repeated dose from the reference and the test product. So I think to me, this it's still it's valid to be generalizable, but I think maybe one day you have a comment, but I think in case there's certain PK parameter that has some correlation, like within the same period, like if you measure 2 consecutive dose, it shows very differently compared to if you measure after switching, you wait to steady state. I think that might be some case we can discuss more, but to me, it's generalizable.
Yeah, so the question being, so when the QMM proposed this model, so they consulted Stat and Don Schumann and I, we looked at this model and we agree that, this can be a valid model for steady state. Change statistics, it doesn't really matter what formulation you have, what product you have, whether it's biosimilar or bioequivalence studies. But one thing I want to caution, which you also alluded, is that so there can be unequal carryover effect between test and reference. For example, from T1 to T2 or 1 to R2. If there is unequal carryover effect, then so further investigation is to be done. So you need to test before you do the bioequivalence assessment, you need to first test whether there is an equal carryover effect between passing numbers. Thank you.
Thank you. Thank you for your comment. So I'd like to move to question promoted. We received a lot of questions. So I may pose a question to myself because I was a presenter. It's a simple question. Is it possible to include unapproved indication based on the safety in that population? So our general response to this type of question is no. We always recommend ignoring patient for B study based on the approved labeling. So that is my simple question. Simple response to that question. So other question, I may pose this question to Dr. Dejan Krajcar. So what alternative or adaptive approaches can be considered to support PK and BE requirement in such a setting? Such a setting means the oncology product B study.
Yeah, thanks for the question. So I think during the presentation we saw today, we got feeling like if we have to go to the background trial on oncology patients, and we have to do everything we can to minimize the number of patients we need. So adaptive designs, as we presented it, for replicate designs with in A, B setting are definitely choice of options that one can take. That's probably one thing when it comes to executing these trials, and then the other side of it is like how to avoid these trials. And today we saw a huge specter of like, first let's address the BCS biowaiver options, and let's see if even if we are violating some of the criteria, could we still justify that something is not in vivo relevant. So I think that's a very important thing that was shown today. And then we can, of course, through the quantitative biopharmaceutics, support this through PBPK modeling, which is a very relevant field that is evolving also because of these cases where we are not able to perform trials. Because, even sometimes you would have a request that you need to perform as per guidance studies on patients, but it is really hard to recruit patients sometimes. So that's where we go and explore other options. So I know I didn't add probably something new in terms of what was presented, but I hope the summary was at least informative.
Understood. So I'd like to ask Dr. Yuqing Gong to make any comment on the question.
Sure. I think for the oncology patient BE study, I think I see because it has to follow the patient dosing regimen, I did see some challenges, or like sometimes people can only conduct like parallel design when it fall under some patient dosing regimen, and sometimes for steady state design take very long time, and it has has high variability. Then it's very difficult to verify steady state based on pre-dose sampling. So I think I see some alternatives like I presented here. It's if you enable modeling, you may have alternative to steady state study. For example, you may not need to reach to steady state. You can have a non-steady state study, then correct for the carryover. So I do see those type of alternative design has the benefit. But I think if the applicant is thinking about alternative design, we encourage them to contact FDA early during pre-development meeting or the MIE pilot, so we can assess those designs in advance. Yeah.
Thank you. Yes, so we really, into the visiting or alternative approach to propose any, the option or alternative approach to conduct the B study in patient study, a patient B study. And I would like to shift the gear in terms of question. I would ask this question, Emilija and Dr. Li Gong. The simple question: can BCS4 drug be considered for biowaiver? Probably I ask Emilija for this question.
Yeah, it's a very provocative question. I would revert back to opening presentation by Rob when he explained the BCS classification as categorical classification and not the continuous one. I like that paradigm. Practically, when we say BCS4, that doesn't mean to be very, very low solubility, very, very low permeability. It may be border of both. So in order to assess whether it's a candidate or not, we can do biopharmaceutics risk assessment. So again, I'm not saying that BCS4 biowaiver for class 4, BCS biowaiver for class 4 is an easy task. But similarly, when we show parameters on the border of the criteria selected on a conservative basis, we can do biopharmaceutics risk assessment, see what scientific evidence is out there to support our thinking and scientific rationale, and we can take it from there. Typically, I would recommend to always engage with the agency for alternative approaches to see whether that is a feasible pathway or not. Thank you.
Okay. Dr. Gong, do you have any comment on that?
Thank you for your questions. But well, right now we for our oncology drug product, we only as alternative BCS class waiver for class 1 and class 3. So I might turn over to for BCS class 4 waiver.
Yeah, Dr. Cheng who is a team leader for immediate release product. So he's going to make a comment on that. Please.
Thank you for posing this question. And my team member, Shamema, she's going to discuss the risk management for the BCS class 4. But for the current questions just asking whether the BCS class 4 is eligible or feasible for the bio waiver, I don't know how to connect the BCS class 4 to bio waiver, and we all know typically BCS class 4 is not eligible for bio waiver. Maybe for the strength waiver, yes, and that's definitely is eligible. And I think for today's talk, I've heard how the industry and regulatory think and the challenges and uncertainties, especially for BCS class 4, because the low solubility and low permeability. But how low is the low? How low is for the permeability? How low is for the solubility? This is very challenging question. And what I think what matters is we discussed about the risk and also the formulation design and the totality of the evidence. The risk is really under the context of N13A and whether there is some risk for the variability for the PK performance and the different GI conditions, and also different type of the meals. And so when we think about this, we look at formulation, then as Rob mentioned, we look at the product similarities. That's we come out Q1, Q2, this kind of scenario. And maybe that similarity can apply to BCS class 4 when it's like BCS class 1 like or BCS class 3 like. We can apply Q1, Q2 or product similarity to those kind of BCS 1 or 3 like BCS class 4 drug substance. However, we still need to consider the specific product design, like the formulation complexity, salt dispersion, all this. Maybe Q1, Q2 is not sufficient because the product design is very specific. They utilize some advanced formulation design. So the excipient may have different functions, roles, even you have Q1, Q2. So that Q1, Q2 is very important element, but it's very essential, but cannot address all the scenarios. The last is the totality of the evidence. So we look at all the data and evidence to support any waivers or any bridgings for some challenge products and complex products. And we really want to balance the science rigor and also efficiency. That's how we are currently moving on.
That's a great answer. And I commend the group on thinking through this. I do have a question about these oncology drugs. So many oncology drugs, your dosing needs to be fairly precise because you're targeting a tumor, you're trying to get rid of the tumor, but you also don't want to raise the toxicity to a level that can result in, say, unnecessary QT prolongation or other aspects, so for example, the kinase inhibitor. So are there consideration for some of these to be NTI or narrow therapeutic index drugs? Have that come up in discussion at all with industry? And if that's a consideration, what sort of things do you have to think about for, you mentioned Q1, Q2 aspects, and how to make it, make sure your dosing is as precise as the RLD?
Thank you, welcome. So we're going to address one last question. Sorry that we, you have a comment? Go ahead, yeah.
Hi, well, the follow-up question to Yi-Hsien. So for the ABB study design, as we know, the agency recommend reference skills fully replicate study design for highly variable drug. So what's your general ideas for those study design, the highly variable drug? And follow-up question is, what's your general idea for the consideration for the carryover effect to the SWR evaluation? So within subject variability. Yeah, thank you.
I think for the AABB design, if you observe high intra-subject variability, then it can be applied to reference scale BE for your first question. And for the second question regarding carryover effect to SWR, I think when you, maybe I can comment first. I think if you have a carryover, like in my presentation, I'm not asking you to ignore the carryover. I think in my presentation, I'm suggesting to use alternative ways like modeling to correct the carryover. So I think we do see if you have carryover, you directly analyze the data, then that will inflate the type 1 error. And I see our biostat colleague, Wang Jie, may want to add a comment here.
So the carryover effect does have impact on the bioequivalence assessment, but it does not impact the estimation of SWR.
Thank you. I'm going to move the left question to Dr. Park. Is a PBPK a potential approach for high-risk product? High-risk product.
Yeah, thanks for the question. This is a similar question to the BCS class 4.
Right.
So the answer is likely to be a similar. Yes, we can do biopharmaceutic risk assessment for those drugs, keeping in mind that a totality of the evidence is more suitable right now because for a high-risk product, they may have several reasons for being a high-risk drug. One of the many could be high-risk formulation design. Right, so that need to be considered, and as of now, probably a totality of the evidence is the best approach, but definitely in future we are looking forward for PBPK modeling being a very integral part of that evidence, and support these high-risk products.
Thank you. So I would like to conclude this session. I'm sorry that we could not address all questions from virtual attendee and floor. So I really appreciate all attendee and virtual attendee, virtual presenter. Thank you so much.
Hello, everyone. And now, welcome to session 2, nitrosamine-driven reformulation and the bioequivalence risk assessment and regulatory decision-making. My name is Fang Wu. I'm a senior pharmacologist in Division of Quantitative Methods and Modeling, Office Research Standard in FDA. I was coordinating this session with Dr. Emilija, Fredro-Kumbaradzi from the Apotex. Now I'm going to serve as a moderator for this session. This session examines how nitrosamine-driven reformulation should be evaluated within BE framework, which include the scientific justifications, risk-based assessment, and when alternative BE evidence is appropriate, particularly for BCS4, immediate and modified release drug products. For this session, we will have 6 talks, and there are 4 talks before the lunch break and 2 talks after the lunch break. We will have 4 speakers from industry and from FDA, and also, and after the lunch break, we will have 2 talks from industry and FDA as well. The first, in terms of time, I would like to welcome our first speaker, Dr. Rajkumar Boddu, who is the team lead for biopharmaceutical from Dr. Reddy's, and to give his talk His talk is decision principles for nitrosamine impacted BCS4 immediate or modified release products. He will be present virtually. Welcome.
One and all. Yeah. So first of all, I would like to thanks to FDA CRCG team for giving me this opportunity. On this note, I would like to start my presentation on decision principles for the nitrosamine impacted BCS class 4 immediate and modified release products. So this is standard disclaimer. The opinions expressed herein are solely belongs to me and does not represent DR opinions. That is nothing but Dr. Reddy's opinions. So this is outline of presentation. First, I would like to cover the FDA guidance and waiver opportunities, followed by BE risk assessment for BCS class 4 IR and MR products. So in this, I would like to cover a couple of case studies. So BCS class 4 IR products, and then I will brief about the modified release product case study, and then I'll talk about some literature review for the excipient impact, and then I'll explain about the workflow for the BE risk assessment, and then I'll conclude my presentation. So coming to the FDA guidance on control of nitrosamine impurities in human drugs. So the guidance specifically highlighted the opportunities to use IV-IVC or physiologically based pharmacokinetic models to waive repeat BE studies for the reformulated BCS class 4 IR and modified release drug products. So can we leverage the PBBM or PBBK molding approaches to avoid repeat BE studies for the reformulated BCS class 4 containing IR and MR products? So to understand how we can leverage the PBBK or PBBM models to get the BE waiver for the reformulated drug product to mitigate the nitrosamine risk, I have taken a couple of case studies here, the edoxaban and bosutinib. So here the objective is to primarily assess the impact of excipients on the absorption. So mainly the permeability and dissolution parameters. So what we did, so here we have developed the model by integrating the physicochemical and biopharmaceutical properties like solubility, permeability, and disposition and transporter kinetics, and we developed the model. Then we have validated the model by utilizing the literature available clinical data. So in this case, we have utilized the intravenous as well as oral data. We ensured the prediction error less than 15% for both Cmax and EC parameters. Then what we did, like so after validation, so we have done the model application. So we have evaluated the impact of permeability or dissolution in the range of plus or minus 10 to 20% range. Then followed by, we have done the parametric sensitivity analysis for permeability as well as dissolution to know the impact on the in vivo exposure. So coming to the case study 1, that is the edoxaban. So here, so as I shown in the previous slide, so the edoxaban is a PGP substrate. So we have evaluated the transporter kinetics impact on both Cmax and AUC. Here, if you see that Vmax and Km of particular transporter did not show any impact on the Cmax, but there is slightly impact on the AUC. Whereas if you see the permeability impact, so within plus or minus 10 to 20% range, so we can see there is no significant impact on Cmax and AUC, but there is slight impact on Cmax. So in the range of plus 20% range, we got around 110% ratio for the Cmax. For in the minus 20%, we got 87% ratio for the Cmax. Whereas in case of AUC, we have not seen much impact. So the excipients used for the nitrosamine risk mitigation may influence the PGP or permeability, thereby affecting the PK parameters, which can be assessed through PBBK modeling. Coming to the other case study, bosutinib. So here also Vmax and Km of PGP did not show any impact on Cmax as well as AUC. So in case of permeability impact, here we can see the significant impact, whereas in the first case, we cannot see significant impact of the permeability on edoxaban, Cmax and AUC. So whereas in the current case, clearly we can see the permeability impact. So within plus or minus 20% range, we got around 65% to 148% Cmax range. So in case of AUC, the impact is around 66 to 144%. Clearly, we can see the impact of permeability on both Cmax and AUC parameters. So coming to the impact of dissolution, here the dissolution has impact. Clearly, we can see the impact of dissolution on Cmax as well as AUC, especially at minus 20% and plus 20% range. At minus 20% range, the Cmax the ratio is around 76%. The AUC ratio is around 81%. In case of the plus 20% dissolution profiles, so we got around 111% ratio for the Cmax. The AUC is around 109%. Therefore, the dissolution safe space will be considered for the target formulation development, and the biopredictability will be validated against the actual in vivo data. So coming to the other case study, that is modified release formulation. So here the molecule is exhibits nonlinearity and substrate for the transporters at absorption level. So we have conducted B study at higher strength, that is 1000 mg. We got successful BE under both fasting and fed. So due to nitrosamine formation, a few alterations were done in the formulation, and we have done the B risk assessment using PBPK modeling. So if you see before evaluation of the reformulated formulation, we have developed the model and we have validated. Here you can see we have predicted the fasting ratios as well as the fed ratios in line to the observed data. So the Cmax and AUC, the predicted ratios are well within observed data. So coming to the dissolution profiles of reformulated product, so in the biopredictive media, which has shown clearly the reduced dissolution of reduced dissolution profiles. So however, that is well within boundaries of clinically relevant dissolution specification. Then, so as an illustration purpose, just I have shown the fed state virtual BE trials. So which has confirmed the bioequivalence for the upper versus lower specification data, which is worst case. The reformulated product is within boundaries of lower and upper specification, and this PBPK model predicted successful BE for the reformulated test products. So in current case, the excipients in the reformulated product may not impact the solubility or dissolution of APS significantly. So maybe in other cases, the impact of excipients on transporter effect to be considered, which can be evaluated through the PBPK modeling. So coming to the literature review, so here the literature review clearly indicated that both BCS class 3 and class 4 drugs are studied for impact of excipient on permeability using various in vitro, ex vivo, and in silico tools. However, specifically for BCS class 4 molecules, so as the excipients may impact solubility, dissolution, together with the permeability, it is important to consider all three factors. So coming to the workflow for the BE risk assessment, based on the in-house experience as well as literature evidence, we have framed the workflow for the BE risk assessment. So here, this is the biopharmaceutics risk assessment for BCS class 4 IR and MR products after reformulation to address nitrosamine impurities. So in this regard, we need to have some in vitro tools and need to generate the data, in vitro data. By utilizing the in vitro data, we need to integrate those in vitro data with in silico methods. Then we should evaluate the BE risk assessment. So coming to the in vitro part, so as I mentioned, so solubility studies and permeability studies, dissolution studies are the part of in vitro data generation. So we should evaluate the solubility with or without antioxidants or pH modifier or any other excipients which are going to be used in the reformulated product. Then we also should evaluate the permeability data. We need to generate the data with or without those excipients which we are going to use in the reformulated product. Similar the case for the dissolution data as well. The pre and post change formulations data has to be generated in predictive dissolution method. So if any of these attributes like solubility, permeability, or dissolution is changed in pre and post change formulation, evaluate the impact through biopharmaceutical risk assessment like PBPK and PBPM modeling approach. So coming to the PBPK model utilized for the BE risk assessment, we have framed the model workflow. So as I mentioned in the couple of case studies, the same workflow we have utilized. So we need to develop the model. For that, we should integrate physicochemical biopharmaceutical properties as well as disposition and transporter kinetics. We should develop the model. Then we should validate the model by utilizing the literature and in-house pilot studies and followed by in-house pivotal studies. We also need to ensure the prediction error less than 15% for both Cmax and AUC. And also we can ensure the prediction error for both pilot and pivotal B studies. Then we should go for the model application. So as we have generated the data for the solubility as well as permeability and dissolution of post-change formulation, we need to utilize those information and we should do the virtual BE evaluation for post-change formulation. And then, so once we get the virtual BE outcome, if at all, if it is giving the positive BE outcome, that is nothing but well within 80 to 125% range, then we should draft the report and approach the agency for the waiver. So this is overall approach for the BE risk assessment. So coming to the conclusions, as I mentioned, US FDA specifically mentions the importance of IVAVC, PVVK molding. So to waive the repeat BE studies, especially for the reformulated BCS class 4 IR and MR products. So in this regard, in vitro assessment of solubility, dissolution, and permeability in the presence of nitrosamine mitigating excipients has to be performed, followed by PVVK molding to evaluate the bioequivalence risk in reformulated products, and then BE waiver can be attempted for BCS class 4 IR and MR products. So coming to the literature review, clearly indicates that significant research performed for understanding the excipients impact on permeability of BCS class 3 and class 4 drugs, which can be utilized for the BE risk assessment. So overall, the PVVK modeling continues to play a pivotal role in supporting BE waiver opportunities, reducing the need of repeat BE studies and accelerating the regulatory approvals. On this note, I am concluding my presentation. Then I would like to thanks to Dr. Shivacharan Kollipara, Dr. Anuj Kumar, and our biopharmaceutics team and DRL management for providing all infrastructure and support. All other CFTs I would like to thank. And finally, I would like to thanks to US FDA and CRCG team for giving me this opportunity. Thank you all. On this note, I would like to call upon Dr. Nasrin, research scientist, OGD FDA, to present on nitrosamine impacted drug products containing BCS class 4 drug substances.
The Shamema Nasrin.
Good morning, everyone. What a great group we have here today. And I can feel the energy in the room, and I hope I can keep this momentum going. I'll just see it now. Thank you, Fang, for introducing me. And before I start my presentation, I will go through a disclaimer. Arshad. This presentation reflects the views of the author and should not be constructed to represent FDA's views or policies. So here is the roadmap for next 10 minutes. We will start by looking at nitrosamine across oral IR product. Then we will talk about why BCS4 is special challenge. I will walk you through a proposed risk-based subcategorization framework for BCS4 drugs. And then we will finish with a real case study that shows the framework in action. So this slide kind of puts the problem in perspective. Looking at the left chart, about 40% of all oral IR products are actually nitrosamine impacted. That is 160 products, not a small number. And if you look at the right chart of these 160 impacted products, almost 20% are BCS4. So that is 32 BCS4 products that's nitrosamine impacted. And we all know that BCS4 products are challenging due to low solubility, low permeability, and also it has a formulation sensitivity. I'll skip this because we don't have time. So what does the FDA guidance say about the nitrosamine impacted drug product? So for the manufacturers who need to reformulate the products to reduce the nitrosamine impurities by adding excipients such as antioxidant and pH modifier. The FDA guidance allows for the use of alternative B approaches to demonstrate B without the needing of the in vivo B study. But there is a caveat, or there is a point. BCS classification plays an important role in determining these alternative B approaches. you can see here that this for BCS1, 2, and 3 class drug, comparative dissolution testing with like multi-pH dissolution profiles may be used as an alternative B approach. If the reformulated IR product uses pH modifiers or specific antioxidants, that should be like within acceptable limits. But for BCS product, the guidance says you need more. So maybe you need like the study should be supported by a validated IVIVC or PBPK modeling or sometimes in vivo BA studies. So why BCS4 is different and how we can scientifically minimize or reduce the study burden. So for BCS4 drugs, we all know that absorption is really challenging because they have both low solubility and low permeability, which means that BA or BE study is quite unpredictable. Because of that, relying on the comparative dissolution testing doesn't always give us a clear picture of what's going to happen in vivo, right? And another important point is the formulation sensitivity. Even small changes in the formulation can have a meaningful impact. So we often need a thorough BE approach to make sure that therapeutic performance remains consistent. So actually, that's why we think the way forward is really a risk-based approach. This means that combining robust dissolution data with the tools like PBPK model when needed. And in some cases, like when the changes involve antioxidant or like pH modification with minimal impact of absorption, we may be able to justify weaving in vivo BE studies. So I think this slide is the key scientific contribution of this work. So the idea here is that the BCS4 class is not like a one big thing, but it's kind of a spectrum. So on the left side, you can see that there are the products, they are kind of solubility limited and they can act like BCS2 drugs. So we kind of listed what can be the risk factor for those kind of products, what kind of food effect we kind of like expect from them, and what are the critical, and identified the critical excipients for those. As usual, you can see on the left side of the table, the solubility limited products, they act like BCS2-like product, and the risk factor can be like pH dependency, it can precipitate, or it has a low solubility and poor dissolution. The expected food effect will be like a positive effect with higher fraction absorbed. And we also identified the critical excipient. And then if you look at the right side of the table, you can see the BCS4 drug that can act like a permeability limited drug. So they can act like actually like a BCS3 product. So this shows usually like no to like negative effect with lower fraction absorbed. You can see the risk factor that it will have low passive permeability, it can have efflux transport, they will have a short absorption window. And then we identified the critical excipient will be like the surfactant or the permeability enhancers. And the problem is actually the middle one where it is like a dual limited products, right? They have the low solubility and low passive permeability. The food effect for those drugs will be extremely unpredictable. So the subcategorization is very practical for those kind of drug or all BCS4 drug because the dominant limitation tells you the risk level and what excipients to focus on. Based on that subcategorization, here we proposed a systemic tier risk-based evaluation framework that will kind of provide a gradual evaluation on a risk level based on the drug characteristics. So the key message here that maybe we don't need to automatically go into the tier 4 and we just say this all BCS4 drug is a high-risk product. So we can follow this subcategorization and then kind of like go one by one to characterize the risk. So the foundation of our approach actually came from the extensive research from our division. So they conducted a research on the BCS3 model drug that showed that antioxidants do not alter the permeability. And BCS4 drug with comparable or better permeability or absorption than those BCS3 model drugs, antioxidants are expected to have minimal or no effect on the permeability. Also, I want to mention that BCS4 drug, where the permeability is not the primary limiting factor, antioxidant is not expected to affect the drug permeability and absorption. Given this complex BCS4 formulation, so for the BCS4 complex formulation, it's also essential to confirm that the antioxidant amount threshold do not interfere with the other excipients or kind of introduce the permeability alteration. So then this is the scientific breeze from BCS3 to BCS4 drug. And let's see if this holds up for the real life drug. So here is the case study. We have an IR antibiotic tablet with multiple strength. It is BCS4 drug. For the nitrosamine impurities were detected in all tested batches of marketed product, which kind of exceeded the accepted intake limit. From the analysis, the root cause was determined that it has the nitrite impurities in the excipient, the manufacturing process condition, and storage condition during the shelf life. So to mitigate this risk, manufacturer reformulated the product by adding the antioxidant. And if you look at the table, the only meaningful changes was actually adding the nitrite scavenger and a small adjustment to the diluent. Risk assessment showed that the product is a low-risk product according to ICH N13 guidance, meaning it has no special formulation or special manufacturing techniques. Now let's look at the drug characteristics. The maximum single dose of this drug is 780. It has a pH-dependent solubility, and you can see that it is highly soluble in pH 6, but in pH 6.8 it precipitated. So according to ICH M9, it was not a high soluble drug. The permeability looks moderate to high permeability. You can see that the food effect is little bit negative to, I would say, the minimal food effect. AUC in fed condition dropped 25 to 20% compared to the fasting condition, and Cmax dropped about 25 to 30%. So this tells us that this drug is permeably limited and it fits right off to our right side of the table of our subcategorization. So according to that, it can be a low risk for antioxidant impact. And if you see that the dissolution data before and after formulation, in both cases, more than 85% of the drug dissolves within 15 minutes. Very rapid dissolution, and the profiles are essentially the same before and after adding the antioxidant. So the antioxidant indeed did not change the dissolution behavior. And now the most important slide, the PK data. If you look at the before formulation table, the TR ratio are already within the limits. Very good starting point. Now, if you look at the reformulation, the ratio are essentially 100%. So I feel like the numbers are beautiful, and the bottom line is adding the antioxidant did not impact the B data, and it behaved exactly like it does for BCS 1, 2, or 3 product as per the framework that we mentioned before. So I will summarize this framework's application potential, or I'll make it short, that this case study kind of proven whatever we proposed based on our scientific data, and it demonstrated that antioxidant additions maintain the B and this supports the theoretical basis of the predictable antioxidant impact. I will say that there are like a lots of like a future research needed. Additional like BCS4 cases. We need additional BCS4 cases to refine and validate this proposed framework. Also, we'll keep generating evidence for an alternative B approach. I will skip. So before I finish, I want to take a moment for acknowledgement. First, I want to recognize Gong. This is actually his science. I'm just honored to be presenting it here on behalf of him today. I also want to recognize our team lead, Zhang, the IR team, also the ORS and DTP2 leadership, Dr. Rob, Lei, MJ, and Dongmei. Thank you, everyone.
Thank you, Dr. Rasmi. So next, we will welcome Dr. Yi-Hsien Cheng, who is a research scientist in the QMM Office Research Standard, FDA. Her talk is on the model integrated evidence to support BE assessment for reformulated nitrosamine impacted BCS class 4 drug products. Welcome.
Thanks, Fang, for the kind of introduction. Good morning, good afternoon, and good night to all the attendees here. I would like to take this opportunity to thank the CRCC Workshop Planning Committee and the Office of Generic Drugs for inviting me to give my 2 cents about how model integrated evidence can support the BE assessment for reformulated nitrosamine impacted disease for drug products. This is the standard disclaimer. The presentation reflects my personal views, not FDA's views or policies. Here is what I will cover in my talks. First, I will give a brief introduction, and then I will go over the workflow for identifying the relevant drug product. And then I will introduce the formulation risk classification framework. I will also provide a case study using the PPK modeling, and lastly, the implication and the summary of this study. As a brief introduction here, BCS4 drug products are known for their low solubility and the low permeability, and the associated complexity on BE assessment. The alternative approaches laid out in the nitrosamine guidance for BCS class 1, 2, 3 IR products are deemed not appropriate for BA or BE establishment. However, the guidance opens a door saying that well-established and sufficiently validated PPK modeling can support BA/BE demonstration following formulation changes for nitrosamine impacted BCS4 oral drug products, which can potentially support a biowaiver. Therefore, this study aimed to develop a PPK absorption model to predict the impact of excipient-induced changes in dissolution and the permeability on PK profiles and the BE outcomes for the reformulated nitrosamine impacted BCS4 oral IR product. So here we begin with identifying nitrosamine impacted BCS4 oral drug products. We look into the 1153 PSGs for oral IR products. We end up with the 247 PSGs for BCS4 drug-containing products. Here I want to emphasize that the BCS classification are qualitatively determined. It's not rigorously or formally designated. We apply 2 filters to these BCS4 drug products. First, we identify the drug products with the negative food effects. So why negative food effects? The negative food effects, which are presented as the AUC or the Cmax ratios less than 0.8, which are unexpected for BCS4 products and may indicate the formulation complexity for BE assessment, potentially more susceptible to reformulation challenges, providing a conservative test case for the proposed framework. Then we apply the second filter, which are 32 products are impacted by nitrosamine. We combine these two filters, we end up with 9 PSGs for B64 drug products, showing negative food effects. And 8 out of these products were further analysis based on the BCS characteristics. Here, based on the prior work that introduced by Shamema, Nasrin, we propose this reformulation risk classification framework for BCS4 products. The purpose of this framework is to guide the BE assessment strategy for BCS4 products undergoing the nitrosamine-driven reformulations. And we put our thoughts on how PPK modeling can support the BE demonstrations using the proposed framework. Here we have 2 key parameters added in this proposed framework. One is the dose number, the D naught, which indicates the solubility limitation indicators. We also applied another parameter, bioavailability, which indicates the permeability limitations. So here I want to emphasize on these are the illustrative thresholds. We propose the D node of 10 and the bioavailability of 50% to provide the consideration and the open for scientific discussions. Here we can see that the dose number is the opposite with the solubility. If we have a higher dose number, then it means we have a lower solubility of these species for products. And we further use these 2 key parameters to subcategorize these species for products into 4 categories. The blue part means the low reformulation risk, means the D naught is less than 10, and the bioavailability is over than 50%. And what we count out with the dual limited means those number is very high and with very low bioavailability. In my talk, I will focus on the BCS2 solubility limited product. We further applying the reformulation risk classification framework. And then we end up with 8 drug products showing different risk profiles. We can see that for the drug D and the drug H are truly dual limited, facing the greatest reformulation challenges. And as I said before, I will focus on the drug A, which is classified as the BCS2 solubility limited BCS4 products with the D node larger than 10 and with the bioavailability around 60% to 80%. Here I will give you a short background of these drug A's. Drug A is an antibiotics and with the moderate to high permeability with an absolute bioavailability of 60 to 80%, with no substantial loss by first pass metabolisms. The food effect slightly delays the rate of absorption, Cmax, but the overall drug exposure was not significantly impacted. Drug A undergoes limited hepatic metabolisms and was mainly eliminated by both renal and biliary pathways. So based on the N13A risk classifications, this drug A was classified as a low-risk drug products, as introduced before. So the PSG also recommend one vaccine BE study. So we end up with developing the PPK models under vesting conditions. We use the IV and the tablet formulations to develop model and further validate with the tablet and the suspension form. After sufficiently validation, we further conducted two analyses. The first one is the sensitivity analysis. We investigate the impact of the permeability path and the dissolution changes on PK profiles and the BE outcomes for tablet and the suspension form. We further applied this framework to conduct the virtual BF simulations to compare the pre-change and post-change formulations in healthy subjects. So here we can see with the model development, we can reasonably capture the PK profiles of drug A after IV infusion with acceptable prediction errors for Cmax and the AUCT. We also use we also extrapolate the PPK model to the tablet dosage form. We also can observe that the developed PPK model recently captured the PK profile after the administration of tablet dosage form. So here we also applied this PPK developed PPK model to validate with another tablet dosage form. However, the results are not presented here. We end up with acceptable prediction error as well. So with that, we applied these PPK models to conduct the sensitivity analysis on the permeability change. Here we can see that the permeability changes substantially affect the rate of absorption Cmax, but minimally impact the extent of absorption, which means the AUCT and the AUC infinity for drug A. And we also observed up to 100% increase or 50% decrease in TEP will potentially cause BE failures for Cmax, but not for AUCT or AUC infinity. However, for the dissolution changes, we observed different trends. Here we can see that the reduced drug release significantly impact exposure, meaning the Cmax and the AUCT, AUC, and the AUCT and the AUC infinity. So if we, if the reformulated drug product will reduce the dissolution release to up to 15% or 20%, we will observe the compromised BE results. And we also propose that the dissolution test conducted in the biorelevant media can help characterize the reformulation risk for the nitrosamine inactive BCH4 drug product. We further use the validated PPK models to conduct the CBE simulations as I introduced before. Here we can see that the post-change and the pre-change dissolution profiles incorporated into the PPK model overlap very well, supporting the BE conclusion. However, we also noticed that there is one step short. So the recommended next step would be to conduct the CBE simulation between the post-change formulations processed by ROD using their respective dissolution data in the biorelevant media, if available, so that we can further support the biowaiver. Then we switch gear to the drug A under the suspension form. Here we can see that as another validation examples, the default PPK model also captured very well the suspension. However, I cannot show the prediction errors of this drug A. We also applied the same sensitivity analysis to this drug A on the suspension form. Here we can observe that similar trends are concluded with drug A, that the permeability change have more impact on the Cmax, but not the AUC side. And here we can see that up to 100% increase or 6% decrease in TEP was potentially cause the failure for Cmax, but not for AUCT or AUC infinity. Again, the same sensitivity analysis results highlights the impact of change in the solutions would have profound impact on the Cmax and the AUC, and up to 15 to 20% decrease in the dissolution profiles may compromise the BE. And this again highlight the dissolution tested conducting the biorelevant media can help characterize the reformulation risk for nitrosamine impacted existing drug products. In summary, the simulated results demonstrate that the default PPT model can reasonably capture the PK profiles of drug A under fasting conditions. The permeability change substantially affects the rate of absorption, Cmax, but minimally impact the extent of absorption, AUCT and the AUC infinity. In contrast, a 20% reduction at each time point of the drug release profile for the reformulated product had a dramatic impact on overall drug exposure, resulting in non-BE for all PK parameters. These findings indicate that the dissolution testing in biorelevant media is crucial for assessing the reformulation risk for drug A product. For the solubility limited BCS2 like BCS4 our product, this is the examples in my talk, the drug A with the of 10 and the bioavailability larger than 50%. The sufficiently validated PPT model that incorporates the comparative dissolution testing in biorelevant media may potentially provide scientific justification and thus support a biowaiver. The PPPK model framework, together with the quantitative biopharmaceutics information, the DNOW and the bioavailability, can be used to support the risk assessment and the BE demonstration following the formulation for drug A, and subsequently can be applied to other BCS4 drug products identified in this study. Last but not least, I want to thank my colleagues from ORS, primarily my team lead Fang, my colleagues Arindom, and the EQM management, Andrew and Lucy. I would also like to thank my colleagues previously from BTP to Gong. With his prior work, we further developed this framework for this study, and the chief for the interactive discussion. I would also like to thank the important feedbacks from the ORS leadership, Lei and Rob, and all people involved in this discussion. Thank you, and I will take any question during the panel discussion.
Thank you, Dr. Cheng, for sharing your work with us. And next, we will have another talk before our lunch break by Dr. Blaž Robnik, who is the group head analytics from Sandoz. His talk name is nitrosamine-driven reformulation in oral drug products from root cause analytics to practical mitigation. Welcome.
Yeah, hello everyone. Thank you for the introduction. My talk today will take a slightly different perspective compared to my colleagues. As a pharmaceutical chemist, rather than focusing purely on bioequivalence risk assessment, I'll approach nitrosamine-driven reformulation from more of a chemical perspective and highlight some less frequently discussed aspects of nitrosamine formation. I'll address key variables and drivers that determine nitrosamine risk and outline how mitigation strategies are prioritized. Finally, in the end, I will touch on how these decisions impact bioequivalence considerations. For products originally developed without nitrosamines in mind, the sequence of events typically follows pathway explained on this slide. Once the potential risk for nitrosamines is identified, it's essential to consider what is acceptable and unacceptable from your product quality perspective. In practice, typically levels of nitrosamines above 10% of acceptable intake trigger the next phase, which is root cause analysis. Ultimately, it needs to be demonstrated that the changes introduced lead to acceptable nitrosamine levels and that all other CQAs remain within the specification limits. However, if reformulation is required, it's important to consider also business implications, since such pathways are time and cost demanding, as they may introduce additional regulatory and bioequivalence requirements. I present today two cases. First one involves immediate release oral tablet, and the second one a modified release oral tablet. I've selected these two cases because of the outcome after the first three steps explained on the previous slide was different. In one case, the risk could be adequately controlled with implementation of appropriate controls, and in the other, contributing factors could not be practically controlled, and ultimately reformulation was required. So on to the first case, this first nitrosamine, we call it NPI, originates from an impurity, which is an API degradation product. This degradation product, PI, contains piperazine moiety, which is known to be particularly reactive towards nitrosating species. As part of root cause analysis, excipients were screened, and microcrystalline cellulose was identified as a major nitrite source. And in addition, the calcium phosphate grade, density grade, was found to affect formulation pH and in turn directly impact neutralization rate. To assess sensitivity of these impacts, we produced film-coated tablets with different compositions varying MCC grade and the calcium phosphate grade. We subjected them to a accelerated stability program and measured several different variables to quantify the contribution of individual material attribute to the nitrosamine formation. Based on the stress data we obtained, we then translated the system to reaction network-based kinetic model and made some mechanistic assumptions. First, piperazine impurity was treated as a process impurity and as a degradant of the API. Nitrite was assumed to be consumed in a nitrization reaction and degrading also into nitrogen gas and water. And NPI, so nitrosamine, was described as first order in PI, so the precursor, and second order in nitrite, while also being pH dependent. Arrhenius equation was used to describe temperature behavior for all of the reactions. On the graphs shown below, we can see the comparison of model versus the experimental data, model being the lines and experimental data are the dots, showing both nitrite levels and nitrosamine levels, demonstrating quite good agreement between experimental data and the model, particularly considering inherently complex nature of solid state reactions. We then ran a set of simulations across different input variables to support definition of control limits. Scenarios in the table on the right are normalized inputs and outputs that illustrate model sensitivity. Probably the most relevant finding was that in all examples where low nitrate MCC grade was used, the nitrosamine level, so NPI, was within specification limit. Another important finding, perhaps less intuitive, is the role of PI, the precursor. It is a common misconception that non-limiting reactants play less of an important role in the overall reaction rate. In our case, since we had up to 2 orders of magnitude different levels in between different API batches, actually the opposite hold true, and this precursor played a very important impact on the final nitrosamine level. So based on the analysis, we were able to set control limits for 2 raw materials on the nitrite, for API on the piperazine impurity, and keep the NPI safely within the limits. The second case is a bit different situation in terms of risk drivers. If the last case was relatively well behaved and predictable, this next one was not. Here we have a secondary amine, which is formulated as a non-proportional modified release tablet. Its observed stability behavior alongside being dose dependent was also highly variable, with levels of nitrosamine ranging from below acceptable intake to up to 3 times above the acceptable intake. Extensive root cause investigation identified several contributing factors, including nitrate contribution from excipients and uptake of nitrate during wet mess processing, particularly at fluid bed granulation step. What was initially puzzling was that control of nitrate in excipients was relatively consistent across the batches. So unlike in the first case, here this was not a dominant driver. Processing parameters such as temperature, time, and airflow rate were also fairly comparable between batches. But what differed were the NO2 concentration in the air at the time of batch production. Environmental monitoring showed that we have significant differences in these levels between batches, and more detailed analysis confirmed that we have clear hourly, daily, and seasonal impacts, as can be seen on the slide below. On the graphs below, reflecting typical urban emission patterns and meteorological conditions. Additional unfavorable characteristic, as already mentioned, was the nitrate uptake behavior of certain excipients, which then effectively acted as a reservoir for nitrite, which could later participate in the nitrosation reaction. This slide shows hourly NO2 concentration profiles measured near the production site for 2 manufacturing campaigns. The upper 2 batches were produced in winter, the lower 3 were produced in spring. And the key message here is that effective NO2 exposure experienced during manufacturing is very batch dependent and can have an important impact on the nitrosamine formation and stability, as we can see on the right-hand side. These fluctuations in hourly NO2 concentration in combination with unfavorable nitrate uptake characteristics created conditions that are difficult to control through material specifications and process adjustments alone. Although recent publications have demonstrated some success of chemical filtration in reducing NO2 levels, in our case, line changes were not feasible and reformulation was in this case required. As we already had quite alkaline pH, we opted for addition of antioxidant of scavenger. We first made pre-selection with pre-formulation studies, but ultimately formulation experiments show that not only type and concentration, but also placement of scavenger is very important. We have selected L-cysteine, the concentration of 0.25%, and put it into the intragranular solution. As you can see on the top right, it produced the best results in terms of nitrosamine control. No impact on other CQAs was observed. Once the compositional change is introduced, the key question, of course, becomes what is the biopharmaceutic impact on previous slide, minor quantitative substitution, such as introduction of scavenger, while proportionally reducing the level of an existing non-release controlling excipient, had no impact on, we showed that it had no impact on drug release. No impact was also observed on pH of the dissolution medium, on the solubility, and on dissolution across different pH. No impact was also observed on permeability, as the latter is being supported by involvement of different transporter systems for the API and the scavenger we selected. In addition to the in vitro comparability package, FDA generally requires reformulated modified release products to be supported by either PBPK or IVIVC models to enable bioequivalence bridging. In practice, sufficient in vivo data may not always be available, or model predictivity can be limited, as was in our case, resulting in the need to repeat the bioequivalence study. As a consequence, then companies are required to reassess the overall business cases. So we propose that in the case of minor quantitative substitutions of non-release controlling excipients supported by the data demonstrating solubility, permeability, and dissolution, bioequivalence bridging could be considered acceptable even without fully validated models. And now to my last slide, to my final thoughts. I hope that I was able to convey at least three messages today. First, that a thorough root cause analysis is critical for selecting appropriate remediation strategy. Second, that mechanistic understanding supported by reaction kinetic modeling can be a valuable tool, although some systems may remain difficult to control without an addition of scavenger. And finally, as a proposal, for well-defined changes such as minor quantity substitution of a non-release controlling excipient, when supported by no observed difference in vitro tests, bioequivalence bridging strategies may be scientifically justified to support waiver of an additional bioequivalence also for modified release products. Thank you.